Two presentations. You'll have to bear with me. You just had lunch, so I forgive you if you close your eyes and wake up in 1.5 hours. I'm serious, because for some of you this will be known territory, and this was a step in what's a low-level course, so I take it low level. So for most of you this will be known stuff. In the first presentation, and I didn't practice it at home before the mirror, seeing how long it would take. So we'll have to see how the timing goes, but I'll simply do it. And if it's on time, I can start with the second after some questions, if you allow me, Mr. Chair. In the first presentation, I'll try to introduce plant genetic resources, because we talk about it as if everybody knows what it is. Where it come from, what it is, where it is, why we need to protect it, and different strategies to protect it. Ex situ, in situ, on farm versus crop wild as in nature. And then in the second presentation, I'll talk about the approach of ex situ conservation. And as you saw in the program, after the break, we'll have in situ, and we'll have Arabidopsis Center, which is more a research collection, and plant genetic resources, if you ask them. But anyway, that might be a nice topic for discussion. So let's start. Very important. Plant genetic resources are the source of world's food. No PGR, no food, no people. This is serious business. And it's nice to have fun and to be pleasant. Well, I don't know. Have pleasant, thank you, meals and conversations, but it is serious business. We have to take it seriously. There are other, every time you do something, it doesn't work anymore. Okay, thank you. There are other objectives, such as the biocultural heritage, right? And if we conserve the old varieties in our country, that is an objective. There's also economic value, very importantly, right? Companies depend on access to plant genetic resources because otherwise they can't breed varieties. But primary is that we are dependent of plant genetic resources if it comes to our food production in the long term. Important message. But then the question is, do we need action to conserve it? Or would it also survive if we don't do anything, like we did the first 10,000 years since we started doing agriculture, right? Everything was okay. So why should we have to do and spend money on plant genetic resources now? And if we need to conserve, how and what? All very essential questions that we always should keep in the back of our heads when making the decisions that we make. Okay. This was a picture I borrowed from Luigi Guarino from the Crop Trust as a pipeline where the plant genetic resources are the starting point for breeding, seed systems, varieties, increased yields, adaptation, climate change, food nutrition, security. So if we start with plant genetic resources, we start with the evolution, right? That was way before we were there. And we could see that all kinds of plants and animals and microbial things started to arise. Here's only the plant part. And remarkably, it's only a very small part that we currently eat. We might extend it a little bit with the algae and stuff. But at the moment, it's only the dicots and the monocots that we consume. And that started about 10,000 years ago. You know the story. We were hunter-gatherers, wandering around, shooting a rabbit and picking some berries. And at a certain stage, not very clear why we settled down and started agriculture. That was a very, very interesting process, what happened there. This happened in centers of origin. And again, an interesting thing, that it happened in different places over the world, independently. Where that came from is not so very clear, but it did. And the result was land races. We call that process domestication, turning wild into not wild, domesticated. That was about 10 years ago, as I indicate. Don't touch anything. Now this thing is not working anymore. How often do I have to tell you not to touch? Oh man, I need technical help again. Yeah. Well, thank you, Nigel. Okay, I got it. So, since 10,000 years, farmers were domesticating, and this weed is wild brassica, and we heard that Lorenzo did his PhD in this topic. But if we see what comes out of wild brassica, and this is only Brassica oleracea, so that's what happened in Europe. The kale, the cauliflower, the sprouts, the terminal buds, the stem, that incredible diversity, and it is incredible, coming out of one species. It seemed that if you simply pulled at one part, it became a crop, right? And well, the Brassica oleracea, or Brassica in general, is possibly the most flexible genus. Since I don't know a good other example, has someone similarly versatile? No. So it is the most diverse genus probably. But I mean, other crops also have different uses. Flax as oil, or as a fiber, or cannabis has different purposes, it seems. And so, okay. So that was done by those early farmers without any scientific knowledge, without any education. They created those crops over the last 10,000 years. And that was quite an amazing change, since we're doing a lot of lettuce work in my GMAG. And if you look at wild lettuces, man, you wouldn't think of eating that. I mean, they have spines, there's dirty lattice kind of sap coming out of the things. So however that turned into the crops that we're eating now, the heads is a small miracle, right? We have theories, and we think we know, or we have ideas, but it's certainly not obvious. If we look at carrots, so on the left, that's the wild carrot. Who thinks of making carrots out of these things, right? So it's quite remarkable what happened. Okay, that happened in a number of places in the world, and the first scientists that systematically started researching this, and I would call him the hero of plant genetic resources, is Vavilov. And he was mentioned before this morning, and he can't be mentioned enough. He sadly died in prison of hunger under the Soviet regime, although possibly with his knowledge we're creating so much food and saving so many people from hunger. So this was one of the pictures in a more sad period. But anyway, he defined the centers of origins, and he defined 8 of them, and all over the world, as you can see. So that domestication took place in many different places in the world, although there are hardly any contact between those still primitive people. And well, possibly you know, and this is not complete, but just to give an indication, your breakfast came from all continents, basically. And it's very important to realize that, and I'll make that point later on. We are completely dependent on each other if it comes to plant genetic resources. But the origin was all over the world. That was Vavilov long ago, and others have done work, including Nigel and his group, to further elaborate on that, and found a number of new ones. But still, the picture stays the same. All over the world, that domestication took place and turned those thin, disgusting plants into the crops that we're currently growing and enjoying. After that happened, it started spreading all over the world. And that was mentioned this morning already. Here we see, oh boy, I had hoped for the speaker's notes on this. Hatshep, who knows the name of that pharaoh lady? But anyway, Hatshepsut, something like that, yeah. And the other is Captain Cook, the famous Captain Cook, who was shipping also plant genetic resources over the world. Already, those historical figures were transporting stuff. They were organizing missions to collect material and bring it home to diversify their gene pools at home or in their colonies, in the case of Captain Cook, right? So that's not new. It has come with civilization that we spread crops around and knowledge about crops. Did I say what it says? Traveled with people, adapted to new varieties. That's also an important point. We can see that some crops were domesticated in certain regions, but got diversified in others because the environmental conditions were different. Also the use types were different. So it's not that only diversity can be found in the centers of origin and domestication, but also in the secondary centers of diversity, whatever the terminology is. But it's not restricted to there. And a long time happened there. I mean, if we look at barley, starting in the Fertile Crescent, moving to Ethiopia, and then Ethiopia truly became very, very diverse, right? So that was a process that continued after the domestication. That went all very well. There were occasional famines that happens certainly in those days, and but somehow those genetic resources were not threatened. There was no problem until we rediscovered Mendel's law and started doing things in a scientific way. We were so successful that we created uniform, high-yielding varieties that replaced the old land races. Concept. Old land races were created by those farmers not really knowing what they were doing or not understanding what they were doing. And that resulted in very heterogeneous populations. Those heterogeneous populations would also survive if the winter was cold, if the spring was wet or late or dry and early, because there was genetic diversity in those land races, right? But what did those scientists do? It selected the best line from those land races and another very good line that had complementary traits, crossed those and selected the best line out of that combined the good traits of the two. But that was only one line, that was only one genotype. I'm now talking about the selfing crops and many of the cereals, which means that that variety that you grow then is just one genotype. If spring is early, it might not produce at all. So you become very vulnerable towards those kind of changes. And we had to start irrigating to avoid, because there was no buffering capacity for the amount of rain that was going to fall. We had to, how do you call, add manure? But fertilize, that's the word, yeah. We had to fertilize, we had to make the perfect bed for those varieties. And if we did, we generated huge amounts of yield. It worked perfectly. And I have a picture in a moment to show you the impact of that approach. But we went from the diverse land races to those very uniform modern varieties, right? You notice that I keep it all very simple, right? But I hope you don't mind. And as a result, we saw that diverse land races that were in fact the raw material for the breeding process were being replaced by the result of the breeding process. So that process was destroying its own raw material. And luckily, and also that was mentioned this morning, some people realize that. We call that, by the way, combined with urbanization, which is also a very important factor. So new land uses, which force farmers to do other things, and no longer grow their traditional varieties. And now climate change, of course, that's hitting very hard for the remaining diversity. Causes what we call genetic erosion, loss of genetic diversity. And that seriously started basically with scientific plant breeding. And that's a bit more than a century ago. Well, I'm getting old. It's nearly 2 centuries ago. Okay, another element in the genetic erosion is the shift from many crops to a few crops. And I mean, if you look at Africa with their traditional varieties now being preserved by World Veg in Tanzania, but the diversity of those crops is enormous, and they would simply disappear because tomatoes are more popular. Yeah. So also there we see a narrowing of the genetic diversity, a replacement of crops by modern crops. And there can be a good reason why those crops were not grown, but we take away our option to be wrong and to change our mind in the future if we don't conserve those genetic resources. So why that shift? You see that the global seed industry likes to concentrate on only few crops, obviously. Easier for breeding, for marketing, and for everything. So those underutilized and marginalized crops are poorly researched. We don't know that much. Difficult to breed. Let's stop. Let's concentrate on tomato, right? That's what happens. Also, the fact that due to that, those modern varieties, and then indicate that you have to make their growing conditions perfect, that requires investment. Irrigation is expensive. Everything is expensive. So we had that, what happened in the Green Revolution, those farmers had to scale up, and the number of farmers went down. And as a result, they had to use the modern varieties that were able to grow under those optimal conditions that they created with money from the bank. So they didn't have that much choice, resulting in narrower genetic diversity. It's all very logical, and it could have been predicted. Luckily, it was predicted. Yeah, and the final point there is the globalization of eating culture. Basically, we all eat the same now. I mean, the Greek salad I'll probably have next week in the Netherlands too, right? So, but the hamburger is a bigger problem in this context. The diversity in our food pattern is decreasing. By the way, if you look at on a country scale, we see that it is diversifying, but that's because we used to eat our local products and we now also eat a hamburger, right? But if we look at it at a global scale, we see that it becomes more uniform because we all eat that hamburger. If you can follow me there. Otherwise, ask a question. Result is that we have very few crops, basically. That genetic erosion occurs, we can see. I already mentioned the fact that they yield better, so farmers will tend to choose for those homogenic crops. But and here's some examples of evidence from the State of the World on Plant Genetic Resources for Food and Agriculture from 1997. To be honest, I don't believe these things. In general, this type of reports are very alarmistic. If there's a problem, it has to be a very big problem, you know. But it reported of the home gardens of Korea, 26% remain between 85 and 93. I mean, there's a decrease of 74%. Wheat in China in 49, there were 10,000 varieties. Now there's only 1,000 varieties. Maize in Mexico, 20% of the local varieties that existed in 1930 still remain. Looks very terrible. In reality, usually it's less terrible. As I said, these type of studies tend to be alarmistic. We did a study ourselves on the genetic diversity in wheat, and basically we didn't see that happening, right? Although all the papers repeated each other, but you can also see it in other cases. Number of insects in Europe, for example, but that's another story. But we also have very good examples of where that narrowing of the gene pool resulted in disasters. And the best known, and also mentioned earlier in this meeting, I think, was the Irish potato famine. That's why all those Irish are in the US now, because 1.5 million starved, people starved in Ireland because of that disease. Everybody grew the same potato or derived from the same type of potato, and they all died because of that disease. Could have been prevented if we would have grown more diverse potato. But one was the best, so everybody wanted to have that one. That's the logic. Other examples, the Bengal famine in India, a disease with a difficult name to rice, causing millions of casualties in 43. I didn't know of this. So that simply happens and nobody tells me. Maize leaf blight in the US in the 70s, when it appeared that all maize was based on the same male sterile line and was all vulnerable to the disease, and livestock had to be slaughtered because there was no feed. It was quite something. But anyway, those were well documented and illustrates the fact that we're vulnerable. The real risk is the one that will be on the next slide in future generation, right? That will cause us to starve, and we should prevent that. Now, by the way, I should mention the banana disease you have heard of, that the Cavendish will disappear. But everybody eats Cavendish. All bananas in the Western world are the same clone. Imagine that. So we need to have another banana that fits in those boxes, and that looks the same. It's that is the situation. So to summarize, genetic erosion is taking place, and it is a serious thing. It has big consequences, and we have reported cases where it had big consequences. In the meanwhile, we also see that the importance of the genetic diversity is increasing. We see that farmers and consumers have new demands. They need to grow new varieties because they're not allowed to use pesticides anymore. Or the consumers want to have their lettuce in plastic bags instead or in a head. All kinds of things are changing very rapidly, and more rapidly than in the past. So the turnover in terms of varieties is increasing. We see that globally the population is growing. It will top off, but it will still continue growing to 10 billion or something like that. And those mouths have to be fed. And on top of that, we get the climate change, right? Causing yields to fail and causing all kinds of problems on that level. Actually, I was expecting that by walking around, this guy would have to move the camera all the time, but it will be.
Don't worry.
Okay, good. Yeah. So, I stole this cartoon. We think we can, but we still have to make that reality, right? This is maize. You might have seen this picture. The open pollinated until 1930, 1935. Then they started doing the double crosses, the hybrids, and the single crosses. And now with GMOs, look at that, it's incredible. And this is not such a manipulated graph where the zero is somewhere in the ground. That's zero. So that's an incredible increase of yield thanks to scientific plant breeding. So you can imagine that that farmer will not use the open pollinated. So we heard this morning already that it was Harlan and Martini who first wrote in 38 a warning that if we wouldn't do things, that things would go wrong. And it took some time, but in the 50s, the breeders started on a larger scale issuing warnings that was picked up by the FAO. Some technical conference between 68 and 72 touched this subject. And we saw the first gene banks being established. I say gene banks here because, of course, Vavilov had already started his collections. The predecessor of IPK in Germany went already out. Some nice stories from the war where they went collecting abroad. There were collections, but they were research collections. They were collections to understand genetic diversity. They didn't have the aim to conserve it, right? So it was in the 60s that the first ones came, and that was mainly in the CG that also were firing were starting the Green Revolution. And luckily they realized that Green Revolution would cause large genetic erosion, and they needed to create those collections to prevent that material from getting lost. We can be grateful to these people, because otherwise we would have been in big trouble. At the moment, the CGIAR is managing a large system of gene banks, conserving our staple crops, and really that is something that I'm very, very, very happy with. Since if I look at the rest of the world doing their genetic resources, that's slightly depressing, but we'll get to that. And the CG is doing it pretty well. Sorry? Should I mention Anna Bennett? Yeah. Yeah, no, you're right. Yeah. I wanted a picture because it was getting a bit boring. I hope you're entertained now. And I chose, no, I chose for Erna Bennett a woman important in that FAO process. But I could also have taken Harlan. I could have taken Frankel, Otto Frankel from. So there are many, not that many, but a number of faces that could have been there. I think initially I also had more than one face, but that required me to tell a story about all of them. And now only Erna Bennett, thanks to our chairman. Okay. Important here. I think everything is important, but anyway, there is a huge global interdependence. I already mentioned your breakfast came from all over the world, right? Examples: cassava, maize, groundnut, beans are domesticated in the Americas and now staple crops, these staple crops in sub-Saharan Africa. Sorghum and the millets, domesticated in sub-Saharan Africa, but now major crops in South Asia and Latin America. And if we look at Brazil, one of the countries that are a bit reluctant to share their genetic resources, or at least in the past, half of their food calories are from rice, wheat, and maize. Well, rice comes from Africa, maize from the Fertile wheat from the Fertile Crescent, and maize from Central America. We all eat stuff from elsewhere. And certainly if you live in the Netherlands, I mean, we hardly had any original genetic diversity. All our stuff comes from elsewhere. There was a nice study by Coury et al. 2015. This is a slightly complicated figure, but that's just the point. What we see is those little segments, which are parts of the world, regions. They're on top, North America, well, whatever. And one part of it to the right shows where the diversity from that region is consumed in terms of calories. They analyze the FAO statistics. And the other part shows where the calories consumed in that part of the world came from. Everything comes from everywhere. That's clearly shown here. There are no thick threads. Everything is very, very diffuse. We depend on each other. I recommend that paper. So, how am I doing time-wise? Okay. Since I think I'm nearly there, surprisingly. If we agree that plant genetic resources need to be conserved, the next question is how and what, but I won't touch too much on the what. How, and we have different approaches. We have ex situ, which means that you take it outside of the place of origin. In gene banks, basically. With different ways of conserving it there, but that come later. And that's directly aimed at conserving and providing access. So gene banks have one objective, or actually two, these two: conserve it and make it available. So they have collections of seeds or living, growing plants in tubes with tissue culture, whatever way it's possible, it's most efficient to conserve that material for future generations. And they have processes regarding, procedures regarding safety backup to make sure that if something goes wrong, they can get it back. Viability testing to make sure that the viability that they're not keeping dead seeds. Phytosanitary stuff, legal aspects, everything aimed at conserving and providing access, right? That's ex situ. Then there's in situ, on farm. Basically the way we conserved material the last 10,000 years, right? Which was very efficient at that time, but since the modern agriculture came, lost a lot of material. Land races, traditional varieties grown for use or for usually niche markets. Some places in the world it's still the most important way of producing food, so I shouldn't be too absolute there. But in Europe, for sure, this is the case. And it is conserving. Obviously, everything that's growing there has a conserving role. And providing access, it is a spin-off. It's very difficult to know what is where and to get access to that material, except within the own network. And okay, in situ and nature is the third approach. And then we're talking about crop-out relatives. Nigel will talk after the break about these approaches, although I guess he will focus on the crop-out relatives. Yes. Those poor land races. But anyway, the crop-out relatives are basically the wild relatives occurring in their original state in the wild, as they did since evolution. And the nature conservation and other natural occurrences hardly accessible. That's the big problem. And under climate change, also under threat, as we can see in niche modeling studies. Again, and I put this slide nearly at the end because it might provoke some discussion. If we look at the three different approaches and at the strengths and weaknesses, I would say that ex situ is reliable conservation, it's accessible, and it builds up knowledge about that material that can be shared. But on the weak side, it's technical, complex, relatively expensive, and limited to certain crops. I mean, we're not talking small herbs, for example. The in situ on farm has a high public relations value. It looks good, those farms. No, and that's serious. I mean, that's there's potential for PPB, the participatory plant breeding. Have farmers involved in selecting the material, and direct use, obviously. Weak side is the low capacity, simply because of the low number of accessions. We've got 2,500 letters in our ex situ gene bank, on a farm, you can't grow that number of accessions. The reliability is low. If the farmer decides to switch to modern agriculture, or if it's a hobbyist, take another hobby, the material might be gone. And it has very crop and geographic limits. I mean, the stuff has to be able to grow where it is conserved. In situ, it's in the original state. It's you can rely on nature protection, and although Nigel's concepts of protected areas ask for an additional layer, in the Netherlands we tend to say a lot of money goes into nature protection. We just monitor and try to provide access and create a safety backup, and they'll do the work, right? And that's a big advantage, I think. And it has quite a high capacity. If we incorporate crop wild relatives in a gene bank, they're difficult. They're difficult to regenerate. The viability testing is very difficult. So you can't have that many populations. Whereas where they're growing in the wild, often there's a lot of them, right? And we could never put that in a gene bank. Those are advantages, but on the other side, the accessibility is low. Management can be complex. And it has, again, those crop and geographic limits. Take your note and shoot me in the discussion, please. So, to me, the question on how should we conserve is together. We all have our own strengths and weaknesses. We have to create synergies between those approaches. And to be honest, I've seen this happening over the last 10 years. If we go back 10 years, the in situ people and the ex situ people were fighting, right? Now they're drinking together. And we're searching for ways to... No, isn't it the case? I feel that the tension has disappeared. But anyway, examples of how synergies can be created: on-farm public relations, very important to raise awareness of the importance of PGR in general for all actors. The ex situ can be a safety backup for on-farm material or for crop wild relatives. In nature is an endless reserve of PGR. The on-farm conservation can do clonal crops, which are very difficult in gene banks now. And basically many gene banks have field collections of those clonal crops. That could be done on farm, I guess. But ex situ communication channels to the international users are much better than from the other actors. So the other actors could use those channels. And in the concept.
So I'd like to go back to the previous slide and start the questions off myself. So Theo is not an expert in situ, so please forgive him. But he says here that in nature is an endless reserve of PGR, and that's actually not correct. Because although it may seem that nothing is being lost in nature, if you look at red list assessments of crop wild relatives, between 18 and 36% of crop wild relatives are threatened with imminent extinction. So it's a fairly high level of extinction. So material is being actively lost through all the different threats that normal wild species are suffering. So there is a slight... What is a statement then? All right, okay. Questions then? Questions for Theo, yes? Sorry.
Thank you. Well, the first one is a comment. Well, do you say that on farm it's a great public relation value? Yeah, you said yes. This word, it was correct? Okay, sorry. Well, I think because it's a great conservation system, and because I don't understand plant breeding without PPV. I mean, it should be a participatory breeding with the farmers. If not, I think it's the only approach in a sustainable scenario. And as well, that's just a comment, an opinion, well, whatever. And the other is the ex situ conservation, which at the end, the conclusion is more or less the same. I mean, the last comment is we should obviously work together, the on-farm and the in situ and the ex situ conservation. But I think the ex situ has the problem that the conservation is not adapted to, and mostly it doesn't adapt I mean, you can keep it for 40, 60, 70 years, the seeds, which in a scenario of a climate change, I think that's a risk, a huge risk that we can't accept. Well, I don't know if accept is the verb, but it's too risky. And I think that an on-farm conservation gives you the adaptation that is needed with the actual needs of the population. And because having seeds that they were feeding, for example, we have a lot of seeds that were feeding in Spain. I'm from Spain during the Civil War. Okay, so the needs of the population had changed a lot, you know. So maybe genetically the genes are interesting, but they are not commercial. These varieties. So what we need is to keep the varieties evolutionating. I just invented the word evolving. Thank you very much.
Yeah, I completely disagree. I'm sorry to say. I think that one, if we talk about conservation, we should conserve. If things change, we lose what it was, right? So the conservation task is about keeping what we have so that we can use it in the future. If we talk about adaptation and that evolution that you call, evolution is a wrong term that takes millions of years and spontaneous. No, I mean, it is not evolution, it's selection, right? And I think that based on the knowledge that we have, it's great if farmers do that and NGOs, please continue, and it might result in very useful variants and very useful genotypes. The reality is that the actual adaptation to new environments, new requirements, is done on basis of scientific knowledge by plant breeders. It's their job, it's their business. They make money out of that. Exactly what you're saying, they're doing on a scientific basis. That doesn't mean that it shouldn't be done elsewhere. Great, continue doing that. You get my support and you get my germplasm if you need it. But it's not that if participatory farming, if natural selection or that slow selection would not take place, that we wouldn't have the ability to adapt to new conditions. For that, we have a lot of knowledge so that we can do that targetedly. But please continue and be enthusiastic, because it is also a source of new recombinations. I mean, the modern breeding has concentrated very much on, well, also the variety registration, the DOS requirements ask for uniqueness. I mean, I could imagine that strategy of having bulks, of having mixtures of lines, would be a completely valid strategy for future agriculture. The road towards that strategy is more or less cut off because of modern breeding and modern production, right? So having the options open by alternative routes, such as the ones taken by NGOs doing the selection of under other conditions, is great, and that should be supported. But don't say that if we don't do that, we are not able to adapt to new conditions, because that's not true, I think.
Okay, so perhaps I can just add an example. I think I don't. Maybe Theo was a bit harsh in what he said, but having said that, I do in principle agree with him. We do need both, and let me give you an illustration of why. If you're the big thing at the moment for any branch of agriculture is being able to cope with climate change. And one thing that really worries me is whether the traditional cultivators of land races will be able to match climate change. Now you can see that if you're a plant breeder and you have access to known genetic diversity around the world, and you have that adaptation at your fingertips, you can pick it out and easily use it. But if you're a particular farmer in a particular area, you're not going to have access to that global diversity. And so it really restricts the material you can use. So you're right, one way of getting around this is for local farmers to work with conventional breeders to actually have access to that broader material and make sure it's incorporated in their local material. So I think, you know, Theo is right. We do need to have.
Thank you. Perhaps it will surprise you, but it's a question. I miss a little bit the expression of the obsolete varieties. I know that you think that you have all the genes in your gene banks, but I'm not so sure. Is that an issue that when we look in the future as well, so that we have also there a task that gene banks have to fulfill to collect what has been already done in the whole time of selection and also since the 1870 when research began and breeding work began. So I never see this, but we were always talking about land races, but this word of obsolete varieties has vanished completely from all the information lists I have seen during this meeting. And when
don't worry. I think that obsolete varieties are a normal part of gene bank collections. Obviously, doing the proper sampling and selection of material to include in the collections is never perfect. And also with your feedback and the feedback of everyone, we might be able to improve. But in general, if I look at the collection in my gene bank, a considerable part is obsolete varieties. Yeah.
Okay, I think we better move.
Second talk, and then I will keep my mouth shut for the rest of the meeting. I can't guarantee anything. Is about ex situ. So we saw those three different approaches. How do we do ex situ? And because as you could hear in the introduction, I managed the gene bank in Wageningen, CGN. I will give examples from how we do things. We're certainly not perfect, but I know how we do things, and I can tell you about that. And a recap, but it doesn't really make sense because it was so recent. But PGR outside the place of origin, directly aimed at conserving. Collections of seed plants, tubes, with tissue cultures, procedures regarding safety, backup, private, blah, blah, blah. Okay. In situ, land races, varieties grown for use, conservation and providing accesses, more or less a spin-off. In situ, nature, crop out relatives, nature conservation, and other natural occurrences, hardly accessible. We heard that just now. Important to realize that we're now going to focus on, look at this, on ex situ, right? And how do we do it? Our clear objective, and I repeat it as if it were a mantra as often as I can, is to keep the PGR available for the next generation and make it available to the current. That's why we're on Earth in the ex situ community. Often we forget and start doing completely other things, but this is why we exist. Currently, the users are crop scientists, plant breeders. Where are the farmers? They're not in this list. I think, that the farmers should be served via the breeders who get the material. The material in our gene bank is usually not suitable for growing out, right? Because it's primitive, because it has been discarded in a long time for a good reason. But it can be used in breeding programs. Breeders should use it, scientists should use it, to study. NGOs could use it, farmer organizations could use it, and then it might be that an obsolete variety in the new world has a value and can be reintroduced. I don't know the examples. There are very few times that old obsolete varieties were reintroduced and are popular again. I do know examples, by the way, but in apple varieties, I mean, those are exceptions, right? We don't serve those users, but we do serve breeders and NGOs and scientists. How do we do it? Basically, 3 steps. We have to choose what to include, so to sample the gene pool. We have to make sure that the stuff is conserved properly, and we have to make that stuff available to the user, right? 3 important components, and I'll touch on those separately. See, here's the first one. Sample the gene pool. Ideally, we should use beautiful optimizing techniques that analyze the genetic diversity and do all kinds of things to get a balanced approach. And we have technologies, we have the collection, the core collection concept that could be used, the diversity trees. So on the level of knowledge, everything is there. We could avoid redundancy between gene banks and create high reliable conservation and full access. Dream on. The reality is another one. The reality is dirty, is opportunistic. The gene banks have where the curators could get their hands on or were interested in, or there were research programs going on in the institute and the material was shifted into the gene bank. So the composition of gene banks in general is very bad. And there's a lot of space to improve that composition. And that will save a lot of money, by the way. I tend to say provocatively that in Europe we spent enough money on plant genetic resources. We just spend it the wrong way. If we would spend it in the right way, we would have the best system in the world. But there's politics and reality between that dream and the reality. Okay, what material are we talking about? Very often it's were the working collections of research and breeding programs of the institutes where the gene banks were located. Also material from the market, the obsolete varieties or the modern varieties that are no longer on the variety list are included in the gene banks. Also collecting missions, and my gene bank, I should keep that separate, but we did 2 collecting missions this year, one in Uzbekistan and one in Albania. You go there and collect material. It's not as simple as that because a lot of paperwork has to be done before you can go there to get permission from the national competent authority, to find a local contact who can go with you. You have to know when the stuff is ready for sampling. So it is very complex, but it is doable and it is done. And of course, exchange between gene banks, causing a lot of redundancy. But it happened in the past, and to be honest, it still happens. I might be forgetting sources, so correct me if I'm wrong later. What types of samples are we talking about? Basically, in gene bank circles, we think of advanced cultivars, the modern varieties. We think of the land races, and there come your obsolete varieties. That's a category in between, probably. So they're not distinct categories. Often it's disputable what category they should fall. And think of the farmers' varieties, traditional cultivars, grown in the country where the gene bank is located or elsewhere, but stuff that can be used as a genetic resource. The wild species, the crop wild relatives, as I mentioned, difficult to maintain in the gene bank. It's better to keep them where they are, but then the access is very low. So we concentrate on the wild crop wild relatives of material that is frequently used. So our lattice collection, 2,500 accessions, consists of 1,000 wild accessions. So that's considerable, but that cerealia is relatively easy to regenerate and very much used by breeders. And research material, that's a difficult category because often it's poorly documented and not very clear why you have it in the gene bank. It was just part of that research institute that you're part of, and they shoved it to it. CIMMYT collection in Mexico, 25% of their wheat is research material. They can't answer me why, but it is. I can imagine that they would include some synthetics that they would make or something like that. 25% of that huge collection. Anyway, I'm not talking about the quality of those gene banks, by the way, that I mentioned, but just as an illustration. Conserve the material. That's the second step after the sampling of the material. How? Storage. Seed collections. By far the best way of storing material. The cheapest. You dry the seed, you freeze it, and you forget about it, basically. Once in a while, you take a little sample to see whether it's still viable, and it can live on for 50 years. We do not know, perhaps 100 years, right? So that's cheap and effective. In vitro and cryo, expensive, vulnerable, sometimes necessary. If you want to conserve the genotype, or it's very difficult to get seeds, or whatever good reasons there are to put it in the tube, might cause it expensive, vulnerable, as I said. The potato collection of SIP, the banana collection of biodiversity. Do you know where the biggest banana collection in the world is? In Belgium, yeah. yeah. Well, actually, the other countries were not too eager to receive the material because of diseases. So to create a vitro collection in a country where there's no banana growing made some sense. But the knowledge they had there and the experience had something to do with the past. That's true. Anyway, cassava at CIAT in Colombia. So there are very large vitro collections, but they're difficult. They're difficult and expensive. Then we have the field collections. Think there of clonally propagated crops and perennials. Fruit trees are predominantly kept as trees in the field. Again, also very, very vulnerable. So making a backup in cryo or something like that is attractive, but labor intensive and often technically not obvious. Now, you'll see the first slide of CGM PGR. That's my gene bank. Just to illustrate what I said before, this is such a seed collection. And we've got lots of racks with bags of seeds stored at -20. All those seeds were before that dried until level of 6 to 8% moisture content. We use the international standard of 15% relative humidity in the room and 15 degrees centigrade, and then wait until the equilibrium with the surroundings is established. But there's some variation in ways of doing that. Then we monitor the germination and maintain these standards. For the cultivars and landraces, it should be above 80%. For the wild species, it should be above 60%. And for those of you who know your FAO international standards well, you will say this is not according to the standards. And then we will have a discussion, and in the end, I'll be right or not. Anyway.
Climate change,
why?
Do it with 50 degrees.
Anyway, the challenge is to keep it stable. So if we're talking about a continued adaptation, not to use the word evolution, that doesn't happen. We don't want that. We want the population or the accession to stay as it is. And that's a big challenge. First of all, we have to avoid it disappearing altogether, right? So once in a while, we take a sample and monitor the viability. That's difficult because often, especially with crop out relatives, we do not know how to determine the viability. We do not know what dormancy there is, whether the seed have to be scratched because usually they're in the soil and microorganisms are continuously eating until whatever happens in reality, right? So that's a big problem. And we should really try to find a solution as a community to better improve the protocols for determining the viability. Basically, no, as the most important step, I guess, is created by safety backup. I do a lot of gene bank reviewing. First thing I want to know, where is your safety backup? Also for on-farm activities, for all activities dealing with plant genetic resources, take a little sample, have it dried and packed and frozen somewhere, because things will go wrong sometimes, and then it's good to have a safety backup somewhere else to pick up and use it again. Our material, I guess that's on the next slide, but we have everything duplicated in a colleague gene bank in Europe and everything triplicated in Svalbard at the global seed vault. But that is on the next slide. Maintain the genetic integrity. That's another component of that maintenance thing. And that's a big challenge because we have to avoid contamination. And that's contamination of pollen or of seeds while cleaning or whatever. We have to avoid drift, shift, and all those genetic processes that are unavoidable. And we have to avoid them, right? If we grow a cross-pollinator out to harvest seeds for the next generation, we know that what we harvest will be different from what we have sown. And we don't like that. In the forages, that's a nightmare, of course. But for all cross-pollinators, that's very difficult. You can increase population size. You can harvest equal number of seeds per plant. You can have very many plants and do hand pollination. There are ways of guaranteeing a better genetic integrity, but then you first have to rob a bank because that becomes very, very expensive. So that you have to strike a balance between the changes in the genetic composition and the costs. And that's a hard choice. Some curators are extremists and want to have few hundred plants. That's not possible. Then we have to, where am I? Right there. I didn't talk about the prevention of infection and diseases, right? But we have to in the avoid of loss. In vitro, of course, other processes are difficult. Some are clonal variation. I don't know too much about that, but changes occur while growing it in the tube. So they have to be grown out once in a while to see whether it's still what it's supposed to be, making it only more expensive, but it's necessary. Okay, regarding the germination testing, this is an example from CGN. What we do initial tests for all material. So I have to be sure that the material that comes in is good. It has to comply to the standards. Everything is tested. But then, and that is quite peculiar to many gene banks, we don't test until 25 years later. 25 years later is a long time. And thereafter, we test every 20 years for the barley and wheat and 10 years for all other crops. With an annotation that, and I hope it appears now. Yeah, basically we follow in the thresholds the FAO standard. We add that if we see that the results of the germination tests are close to the threshold, we reduce the term. So if it's just 85, 87%, we will do the next generation viability test after 5 years. So that it becomes more or less a sequential seed testing protocol. We're still testing how it works out, analyzing, doing double blinds to determine the reliability, but it seems to be working. I could say more about that, I won't now. If on the other side of the threshold, it's just below threshold, we don't make it an urgent regeneration. So we have 4 states resulting from a germination test. Either regenerate immediately because the germination is very low, or regenerate within the coming five years. Do the next germination test in 5 years, or follow the protocol of 10 or 20 years.
8 minutes.
Whoa. Okay. I like that. Being sped up. Okay, that we did that because of an analysis when our gene bank was 25, 30 years old. We analyzed all the germination tests that we had and looked at the changes over the period. On the x-axis, you can see the time it was in storage, and on the y-axis, you can see the change of germination. And basically, we didn't see anything. And of course, the increase of germination can't be higher than 10% because of the threshold, or was never higher than 10% because of the threshold going in. So we had 1 or 2 that after 5 years already had critically low big jumps. But in principle, seed doesn't die in a gene bank if you properly store it and you go in with good seed. That's basically the message I got from this. But we do not know what happens after 25 years. There might be species that behave differently, etc., etc. Don't quote me if you see your gene bank being dead, right? I don't take that responsibility, but in principle, it's not the biggest concern. Everything is duplicated in the colleague gene bank, triplicated in Svalbard. Standard routine, don't think, do, right? This is how we handle seed lots. We have one seed lot, which is the base sample, which we keep as long as possible. And we do germination tests on that base sample only. And when the germination goes down, we regenerate. If we run out of seed, because we distribute a lot, we will do user regenerations. We create separate lines that are regenerated just for distribution. But that baseline should be as long as possible, because every regeneration will cause a change of the genetic integrity, right? That's our strategy. I think it's the strategy, but if you have better ones, tell me. Then we have to create fresh seeds. Effective population size I mentioned, the pollen and seed contamination I mentioned, phytosanitary test to determine whether the quality is high enough, and viability test, and then it goes into the gene bank. So at CGN, we test the phyto before it goes into the gene bank for all known pests and diseases that are current at that moment and relevant. I can give details if you like. We do that regeneration in collaborating with the breeding companies. Nearly more than half of our material is regenerated without cost by breeding companies. It has all kinds of advantages, also some negative points. I can elaborate if you like, with my 8 minutes, now 7 minutes.
Now 5 minutes.
Does not allow for that. Of course, isolation of cross-pollinators and so forth. Then make it available. Have good material in terms of quantity and quality. Do collect as much information as you can. That's important, because saying I have 2,500 lattice accessions doesn't help the user. Yeah, he or she has to know what traits are in there, how it can be used, etc. So that data has to be collected and made available in a user-friendly way to that user. Easy requesting. As you can imagine, everybody has to sign an SMTA. That can be done via click wrap without any paperwork, very smoothly, very easily. Also for the search of material, good interface is essential. Presenting the data, providing tools for selecting material, etc. A lot has happened there. You don't have to invent the wheel. The tools have already been developed, I think, to a certain extent at least. Also, the characterization evaluation is done in collaboration with the breeding sector. Characterization we do ourselves. That's during regeneration. Evaluation often requires specific expertise available at the breeding companies. You notice I'm speeding up, right? the value of plant genetic resources is a function of the available information. Obviously, you have to have the seed, right? Without the seed, value is 0. But from that point on, data, that determines the value. And we see that in most gene banks, if not all, documentation is a bottleneck. Not all. And I'm looking at Stefan, and I think IPK and CGN has it pretty well organized, but very expensive. And you're shaking your hand. I could shake my hand too. It's not perfect neither. We're working on it. There is an open source and generally usable solution called GrinCE, Community Edition. I don't have experience with that, but it seems to work. So that might be a solution. If you have a problem, have a look at this solution. I will not use this opportunity to show you our documentation system and interface, but I do invite you to have a look, because we like to have visitors, and it is quite a nice interface, I think. That young guy there is me. Good. Then we start distributing. 4 important questions. Who do we distribute to? How much material? If a Korean scientist sends an email message saying, send me all your letters, do we send them 2,500 accessions? I didn't think so, right? So, phyto, how do we deal with that? Legal, how do we deal with that? All difficult questions. Our policy is the following, and I think I'm approaching the end,
1 minute.
Mr. Chair. Oh no, I don't. I'm still very far to go. These are the principles. We don't send more than 50 samples unless there's an agreement with the user, bona fide users, etc. Possibly this all accessions are without Q and RNQP. What is RNQP? Regulated non-quarantine pests. Thank you. Anyway, and all material is distributed for free. 1 minute. This is the distribution numbers from CGN. You can see half goes to public, half goes to private, half stays in the Netherlands, and half goes outside. We distribute last 10 years on an average 5,000 accessions per year based on that policy. That's disease-free and maximum 50 per user. If we look at the global system, we see very many gene banks. We see a nice centralized documentation system, but the quality varies. And generally, it's too low for effective collaboration. And previous sessions, we already discussed this. If we look at the global system, I think we can say that the major crops are pretty well covered. Crop wild relatives, clonal crop are weak spots. And there's a large redundancy in the major self-feeding staple crops. Rice, wheat, barley, everything is the same everywhere, but they're cheap to maintain. If we look at the European situation, the coordination is weak. We do have ECPGR, but ECPGR is small and doesn't have funds, but it provides a good basis, and we're using that basis. CGIAR, and that's of course worldwide, is an example. They're well coordinated and well managed. Those are good gene banks, and that's very nice to know. There are other gene bank activities, Mr. Chairman. I'm sure you're very interested in that. Gene bank research, we have to improve the efficiency of our activities and services. So there's research needed to do what we need to do. And of course, we have to be able to communicate with the users, participate in their research activities. So we have to know the terms. We have to listen if there's a presentation on bioinformatics or genomics, right? Because we have to be able to interact. That's very important. We are involved in the policy advice. Policymakers need support in the international fora. And it's also our duty as experts to inform the policymakers of the consequences of the policies they're making, because they don't know. We are the experts, so we have to communicate that. Quality management is the way to assure that the quality is an appropriate level. I got quick slides to illustrate these things. This is my biggest achievement in my career, a Nature Genetics paper. Would have loved to talk a lot about that, but that's not the opportunity now. We do support, we are usually part of delegations in international negotiations in the COP from the CBD, in the governing body of the International Treaty, and in the Commission on Genetic Resources from the FAO. And we publish about the consequences. As I said, these are two recent papers about the consequences of ABS rules and DSI options on gene banks. A quality management, we're the proud first gene bank in the world, as far as I know, that were ISO certified for a quality management system. We were forced to, by the way, by our government, because we became a legal task. But now we're very happy that we were forced to. And this is my last slide, Mr. Chair. Perhaps the most important one. We have to act. The threats due to climate change are not sufficiently acted upon. The low access to plant genetic resources, thank you, Mr. Politician, is causing that we can't sample can't collect anymore, and we can't conserve it properly. And we know that the climate crisis will cause further degradation. This is very bad. This is actually terrible. And on the other side, because of the low access, the plant breeders have difficulties creating the varieties that we desperately need in these times of climate change.
Okay, so Nikolai Vavilov did his PhD in St. Petersburg, and he finished it in about 1912, and then was working as a postdoc in one of the universities. And he was working on food security and genetic resources very early on, before really it became publicized. And 1917, the revolution happens in St. Petersburg, and everything's a big mess, basically. The communists take over. First the Mensheviks, then the Bolsheviks come in, the Bolsheviks take over, and because of the chaos after the end of the First World War and the revolution and the Civil War all happening at the same time. There was a civil war because obviously all of the Russians didn't want to join the Bolsheviks. So there was this civil war going on. Three things, all of them caused mass hunger. And so there was a big shortage of bread. So everyone had to queue up each day for their national allocation of bread. And so the Bolsheviks were seen as true communists, equal with everyone else. They queued up for their bread as well. So you had these very long bread queues, and in the bread queues was Vavilov queuing for his bread, but a few people behind him was Trotsky queuing for his bread as well. So he basically slipped back in the queue, and in the 3 hours he was standing next to Trotsky waiting to get his bread. He convinced him to give him the money to set up the Vavilov Institute, his own institute. And remember, this was a young postdoc who got this funding. So this was all fine. The Vavilov Institute was doing absolutely wonderful work early on, and helping secure food security for the Soviet Union as it was over time. But unfortunately for him, the person who actually decided who actually gave him the money initially was Trotsky. And if you know your history, you'll know that Trotsky fell out with Stalin. So when Stalin took over, he sent secret agents, you know, much like the secret agents who came to London recently and killed that Russian dissident, one Russian dissident, and then tried to kill another one and his daughter. So they're still doing the same thing as they did all those years ago. But anyway, that's an aside. So they sent a killer to Mexico City, where Trotsky, who'd thought it was too hot and had moved to Mexico City, and was actually having an affair with Frida Kahlo. If you know Frida Kahlo, the famous painter, was having an affair with her. Anyway, so they came and they killed him with an ice axe that you use for, you know, when you're having your cocktails, if you break up the ice cubes, you have an ice axe for not the ice axe for mountaineering, but the ice axe for breaking up ice cubes. Okay, they kill him with the ice cube, kill him with the ice axe. And then back in Russia, unfortunately, Vavilov is tainted by having had this previous relationship with Trotsky and being seen as a friend of Trotsky. So he was over time, he was ridiculed, and Lysenko, if you know your Russian history, was a sort of another kind, alternative geneticist trying to put forward Lysenko's theory. It was a sort of unadaptability as a false evolutionary concept. But Lysenko was a friend of Stalin. And so, unfortunately, Lysenko told Stalin to get rid of Vavilov. Vavilov was put in prison. So he slept apparently every day with his suitcase packed by the front door. So, because he thought he was going to be arrested. And before they came to arrest him, he picked up his, as he was going out the door, picked up his suitcase so he could take it with him. And they put him, ironically, they took him to a prison in Saratov, which is in the Ural Mountains. And by some very weird coincidence, his wife was from Saratov. So after her husband had been arrested, she wanted to escape the political fallout. And so she went back to live with her parents in Saratov, not realizing that Vavilov, as Theo was saying, was actually being starved to death in his prison cell in Saratov. So he died probably just a few miles from where his wife was living. And it really is a very, very tragic ending to someone who was the founding father of genetic resources. And even more poignant, I'd like to say, is Theo very kindly showed that image of mine from the work we did on looking again at the Vavilov centers. And what was amazing in doing that reanalysis of the Vavilov centers was how much Vavilov got right. When you think he published the first paper in 1926. So he was doing the work between sort of 1918, 1926, and he got such good data sets to be able to do that. So it really is a mammoth achievement. The Vavilov story, or one of them anyway. Yes, it's a very sad story, and it's not nice, but it is an interesting story how these things work out. Okay, so I'm going to start off the conversation on in situ plant conservation. And normally I deal with students, and I always give my students publications on useful websites. Now, I know you can't read these, but it includes lots of additional information that you can download from the internet and will be used to anyone who wants to actually use it. And I'm assuming that Panos, that these presentations will be made available to everyone after the meeting, yes?
Yes, except those that we don't want.
Yeah, so in my case, I do want, so you will be able to get hold of these. So there's no need to take, if you were going to take photographs, don't take photographs. Okay, so let's start off with this image on the left-hand side here, which you can't see, and I can barely see, although I'm only a few millimeters away from it, is my model of how we do plant genetic resources. And basically, we start off at the top with biodiversity. We do this bit at near the top, which is in that big bracket you can see, and that's conservation planning. And then below conservation planning is conservation implementation. And then at the very bottom is moving into utilization. So essentially what we're doing is we're taking our biodiversity, we're deciding what we're going to conserve, we're then planning that conservation, deciding to do in situ conservation. So we go down the right-hand side of that diagram and we go into the 4 basic techniques for in situ conservation. And then we have the products of that in situ wild conserved species or land races conserved on farm if you're doing on farm. And then that material can be characterized and evaluated. So in slightly larger writing, you can see the 4 techniques. So genetic reserve is for crop wild relatives, and it's within a protected area, within the existing protected area. Why do we do conservation within existing protected areas? Because they are already doing conservation. As Theo, you should be up here giving this talk. You said this as well. Theo told you already that they already have a conservation ethos in protected areas, so it's relatively easy to get them to add additional species to their conservation monitoring programs. So the important thing is not management, but monitoring. And if they're actively monitored, then you can check whether the population is going up and down. And if it's going down, then you can correct the management to make sure it's better. And if it's constant or roughly staying the same, then it's basically okay. But the important thing is you choose the site because it has healthy populations already, has large populations of your target taxa. And if you're choosing them for that reason, then you know the management is already correct. There wouldn't be healthy populations, logically, if you think about it, if you didn't have the right sort of management. So ideally, if you're choosing the best sites, then you'll do that. The other kind for, so next we have on-farm. This is the conservation of land races on farm, and I'm not going to go into detail on that. But if you ask me to choose, which I think are the most important elements of plant genetic resources to conserve, I would say land races, interestingly, not crop wild relatives. Why? Because land races can be used by breeders themselves. They can be used by all breeders. Whereas crop wild relatives are a bit more difficult. You have to do some pre-breeding, and not everyone has access to the right techniques to use to incorporate crop wild relatives in their breeding programs. So I think practically, we should have an equal focus on both. So we shouldn't just think crop wild relatives are difficult or land races are difficult and focus on the others. Then right at the bottom here, so in the UK particularly, and I think this process is starting in other countries as well, with protected areas, protected area managers are getting a lot of stick from the government saying this is extremely expensive, having this network of protected areas, because they all require staff to do the conservation. And so they basically want to cut them down. They want to reduce the number. And in reducing the number, they're now looking for alternatives to protected area-based conservation. And so I thought, well, an alternative to this would be what I call extra protected area in situ conservation. It's a long-winded way of saying doing basically in situ protected area genetic reserve type conservation, but outside of a formal protected area. Yes, so you're applying the same management monitoring, but it's not done within an existing protected area. Now, this is interesting because if you look at the habitats that crop wild relatives are found in, they're nearly always disturbed anthropomorphic environments. So they're found on farms, around the edges of fields, in orchards, roadsides, disturbed habitats. Whereas most protected areas are set up to conserve climax communities of really established communities. They're not the sort of things that crop wild relatives generally are suitable for. So then I came up with this idea, and I don't know whether IUCN stole my idea or not. I'd like to think they did, and perhaps gave me a bit of credit, but anyway, they didn't. So they came up with their own idea, which is exactly the same, but they called it Other Effective Area-Based Conservation Measures, which is exactly the same thing. It's doing the same kind of in situ conservation, but outside of protected areas, in non-formally conserved areas. Okay, so then we look at definitions. So we have definitions of in situ. Basically, what we're talking about is the location of our population, our target population, designation of the site for active monitoring or management, and then actually doing the management and monitoring. Though I stress again what I just said, in that you don't necessarily have to do a lot of management, because you're choosing the site because it already has large populations of your target taxa. We have the definitions of the different techniques, but I'm not going to go into them in detail because I'm going to focus for the rest of the talk on genetic reserve conservation, which basically genetic reserve conservation is probably the most widely used in situ conservation technique at the moment. And it focuses on locating the population, managing, monitoring them, and we're focusing on genetic diversity. So we're not focusing on, as normal biodiversity conservationists do, on species level or habitats. We're focusing on genetic diversity within the species, and we're dealing with natural wild populations within defined areas designated for active long-term conservation. So active conservation means we're actually monitoring the species we're trying to conserve. If it's for crop wild relatives, we're monitoring those crop wild relative species. And it has to be long term. You don't want to set up a genetic reserve and then 5 minutes later they're going to build a motorway through the site or something. So it has to be a site that you know is going to be there. You have to have done some climate change analysis to, again, know that the population is likely to exist at that location for the next, say, 30 years, if possible. Okay, so then moving on, we have precise methodologies for how to do both genetic reserve conservation and on-farm conservation. I'd like to go through both, but we don't have time. So I'm going to focus on the crop wild relatives, as I said, within genetic reserve. So the rest of the talk is going to be focusing on going through those points there on the left. So before you get to specific working on-farm, you have to select your target taxa. How do you select your target taxa? There are lots of different criteria you can use. So current conservation status, is the species already conserved in situ? Is the material in the gene bank? If both of the answers is yes, then you don't do it. You don't spend any more money on conserving it additionally. Yes, because it's already well conserved. Socioeconomic use. So for us, crop wild relatives are important because they have economic value, potential economic value. And you've probably all seen that figure of 180 billion per year generated by the use of crop wild relatives in breeding. So it's a huge figure, the amount of additional income that's generated by using crop wild relatives in breeding, not to be underestimated. But there are other means of value as well, other than just straight economic. Whether the actual species is threatened with either extinction or genetic erosion, whether it is very distinct, it has a large number of unique alleles, whether it is found just in one habitat or multiple habitats. Obviously, if you have a species which is just found in one habitat, then it's going to require more conservation than a species which is a generalist that's found in lots of different habitats. Biological importance, you know, in any particular habitat, some species dominate. Usually it's trees. Out here it's eucalyptus, which is a non-native, which we should tear down, Panos. You should get someone out with a chainsaw and cut them down and plant a nice native tree outside your window. But biologically important species, those need to be prioritized. Culturally important. So usually most countries have a kind of symbolic plant that is important for them. So there's a lot of money invested in their conservation. Like you think of Lebanon, you think of the cedar of Lebanon. The amount of money that's spent on Lebanon on cedar of Lebanon conservation is huge. Species mentioned in legislation. In Europe, the Habitat Directive, the Bird Directive, both list plant species which should be conserved. How costly it is to conserve a species, ethical reasons for conserving a species, aesthetic reasons. A lot of money goes into conserving orchids. Why? Because the general public like orchids. They're pretty. And if the general public like them, then they're going to vote for a party, hopefully, that says they're going to spend money on conservation or conservation of orchids. And then the priorities of the conservation agency. So all of these different factors can be used, but the ones I think are the most important ones for us are the ones that I've shaded in green here. Then once you've sorted out your target taxa, then you have to do some kind of ecogeography gap analysis, which is basically collecting ecological, geographical, genetic, taxonomic data, and then using basically GIS analysis to predict where those species are found, and finding, usually for crop wild relatives, where multiple species are found. So very rarely do we set up a genetic reserve just for single species. What we're trying to identify is sites that have 10, 20, 30 crop wild relatives all growing at the same location, so that you get a lot of bang for your buck, as they say. And then out of that, out of your gap analysis and ecogeography, you get your conservation objectives. So if we take this species here, this is Bavilovia formosa. Can you just about see it? It's the wild relative of pea, garden pea, and it's found in the Caucasus over 2,000 meters, growing on limestone scree. And although you can't see it from this image because it's a close-up, the mountain is sort of like this. It's extremely steep. And it also has very, very long roots because there's a lot of scree before you get to any soil. So it is very, very hardy. And so we've done our ecogeographic survey, we've done our gap analysis, and we found that there is no active conservation in situ and no material in gene banks. There are about 16 seeds that were said to be in the Vavilov Institute, but we tested them and all of them would perhaps I shouldn't say this online, all of them were not well. So what do we do in terms of conservation? Well, we don't know how to bring them down. There's obviously some reason why they won't go into gene banks and survive. And maybe it's a sort of relationship that Theo was talking about previously, where they have some macrobiotic or microbe relationship that we don't know about. But anyway, it doesn't work. So what we need to do is set up protected areas on those mountain tops, and that I'm happy to say has been done in Armenia. If we don't have all the data we need, then you need to do some kind of field exploration. You need to actually go and look and collect additional data so you can work out your conservation priorities. And if you do have enough data, it'll tell you this particular location has 20 species of crop wild relative growing here, but you need to go and test and actually check that the 20 there, the background data you've got is correct. Okay, so let's move now into the in situ model. So the first one is site assessment. You've got a lot of potential sites to choose. So you want to choose the ones that contain the morphological and genetic diversity of your target taxa, multiple crop wild relatives in this case. And you want to actually make sure those habitats contain ecogeographic amplitude. So what we mean by that is different habitats, lots of different habitats, some at low altitude, some at high altitude. So they represent the full range of those species you're trying to conserve. And obviously you don't just set up one reserve because if you're taking ex situ samples, you sample from multiple sites for each species, yes? And the reason for doing that is you don't find all the genetic diversity in one location. So you can't have just one in situ site. You have to have multiple sites. And the geneticists tell us that we need at least 5 sites for each species. And the actual examples of where you have the impact of this assessment of socio-political. So there are various constraints in setting up sites. We don't have the money to buy new sites. So often we're working with the government to try and get them to give us the site. And so often the sites they want to give you are not ideal. So for instance, some work I did in Syria, this castle here is Khalat Saladin, which I can't tell you a story about because we haven't got time. Basically, it's a very famous castle for the Arabs. And right beside it is this area at the bottom, which was, I think, possibly one of the first genetic reserves actually established. This was set up by Akada, which was also based in Syria. Now it's moved to Lebanon and Morocco. And why did the Syrian government give us this land to actually set up the genetic reserve on? Because it's covered in unexploded bombs. So the local people don't go in there. But if you're careful, and you have to be very careful, you can actually go in. It's absolutely jam-packed full of crop wild relatives. So it's the perfect site. No one's going to come in and graze their animals there. Because of the bombs, and it works, yes? So what I'm telling you is a rather silly story, is to make the point that we very rarely have ideal locations to set up our genetic reserves. We often have to negotiate with the ministry, and it's difficult to get the ideal sites. Having said that, in the UK, the best site is the first genetic reserve that was set up, but I'll show you that later. So there is not one simple approach. Often they're associated with existing protected areas, I said, and it depends very much on the country. Then once you've actually identified your site, then you need to basically set up this standard pattern where if you look at the diagram on the right-hand side, you have a core area of your protected area of your genetic reserve, which contains the minimum viable population. And minimum viable population depends on the species. The number of individuals you need to maintain genetic integrity depends on the species, will vary to species, But if you ask the geneticists, really push them, they'll come up with the figure of 5,000 individuals. So what you're looking for as a minimum is 5,000 individuals in the black area in the middle. And that's where you focus your management and monitoring techniques. Then outside of that, you have what's called a transition zone and then outer zones. So the transition zone is you have some management, but it also has populations of your target taxon. So you can get immigration between the populations inside and outside the actual genetic reserves. So it just helps sustain genetic diversity over time. It's expensive, as I said, to set up a protected area. Most protected areas have some kind of staff involved with monitoring it. And the taxon has to be suitable to be maintained in one location. One of the species we're trying to do this for is wild lentils in Uzbekistan. And with wild lentils in Uzbekistan, you find a population in one location, and they're very small populations. You may have a population as small as this area of carpet here, and it would have, say, 10,000 individuals. So you get a lot of them in a mass together. And then if you go back, interestingly, next year, you could find none. So it is actually quite difficult. So you need to have a large enough area so you're getting a coverage of those species year on year. So you have to, if they do move around, then you've got it included in your genetic reserves. And it has to be sustainable. It has to be non-impacted by climate change in the short term, and it has to be not threatened by a development project or not threatened by building a road or something. Then we're looking at what is a viable population. Well, a viable population defined by IUCN is just the population that can maintain genetic diversity for the long period. And IUCN actually talk about 1,000 years. I think we would seem that as rather ambitious to try and actively conserve something for 1,000 years. So I toned it down a bit. I was talking about 100, but I mean, even then it was a guess. And then once you've done that, you can formulate some kind of management plan. So the management plan is just a document which lays out how you're going to actually actively manage the site. Are you going to graze it? If you're going to graze it, with what kind of animal? Because if you don't graze sites, if you don't maintain the current management practices, then basically you find succession will happen. And particularly if you don't graze it, then you'll find trees, you'll find shrubs, then trees coming along. And then that changes the whole taxon complex. So your target taxon may actually be lost. Very good example of this is with Zeodiploperennis, the wild relative of maize in Mexico. So they found the populations in 1956. They literally built a wall around the populations to protect them from everything. So they built a wall. Inside the wall, you have succession. You had trees growing up after a few years. The Zeodiploperennis doesn't grow in the shade. It has to have light. So the populations decrease. So then they had to take the wall down. And it was only once they took the wall down that the populations came back. So it's a very good illustration of the importance of knowing the right management for your site. But usually you can see that by just observing the conditions in the location you're looking at anyway. So you formulate your management plan, which actually states how you're going to manage and how regularly you're going to monitor. Then, as I said, we have definitions here of protected area and genetic reserve. So it's rather a technical definition, but I'll just read it out. A genetic reserve, an area of land or sea, because remember we were talking about animals as well as plants, so it could be fish, for instance, especially dedicated to protection and maintenance of genetic diversity of natural wild species within a defined area designated for active long-term conservation. So it's a variation on the protected area definition that's given above. If you look at IUCN, they group in terms of the protected areas they recognize. They have a whole range of different definitions here, about 7 different types. None of them exactly fits with the genetic reserve for crop wild relatives. Now, I hope no one from IUCN is listening to this, but basically, I've tried for years to convince IUCN to add in an 8th category, which would be a definition of a genetic reserve that would be useful that we could use. So none of these current definitions exactly fit, and IUCN, it's not a priority for them to change to add in for us. But I mean, basically, we can use the definition I've just given. But I'm just making the point that the current definition is not applicable. Then you have your associated with your management plan. You apply it. As you can see at the top, you apply your management plan. You then monitor your progress. And if the population is decreasing, then you go into the orange blobs and you actually revise your management plan, and then implement the revised plan. If the population is staying about the same, then you go on the green circle, going round and round and round over time and doing the same kind of management. And then basically you have a similar structure for reserve monitoring. So you have the monitoring of the site, you have the how the monitoring is done, asking such questions as which taxa are you going to monitor, how often are you going to monitor, et cetera, et cetera. And then you store the data, and then you're looking for those changes in either population size or genetic diversity over time. And usually it's monitored using some kind of quadrat, and here you can see some quadrats we were throwing down, or I took a picture of, throwing down in Palestine. This is on the West Bank. And very interesting, they're supposed to be done at random, the quadrats. If you notice the picture here, the quadrat is got the only area that has a meter by meter square, just happens to have randomly come out. There's no rock included in this site. So you really must obey the methodology. It should be random, not directed as it has here. Okay, so coming towards the end of my talk, once you've done your in situ, then basically the material must be used. You can link it to utilization of local people. So this shot is taken in Jordan, and here we were setting up a genetic reserve in Jordan, and we were working with local farmers. So these are essentially scientists from ICADA and local farmers, and we involved them in the process. And very interestingly, you think farmers may be reticent about having crop-wide relative conservation, but because if you explain to them that crop-wide relatives can be used in maintaining genetic diversity for agriculture and breeding new crop varieties, immediately they get it. It really is amazing. So they are very, in my experience, very, very supportive. So as well as the traditional users, the farmers, you have professional users as well, also exploiting the material, taking the material out for use in breeding programs. And then this is you probably heard my talk yesterday, I hope, and I went through this diagram in detail. But this is just again to stress the importance of making the link between in situ and ex situ. We can't have in situ conservation, in my view, unless it's linked to ex situ, particularly because ex situ provides the link to the user. If we want in situ to be comparable with ex situ, we have to meet the user requirement. Otherwise, it's a pointless exercise. It becomes really academic. Last slide, just to say that what are the products of this? Well, we have living plants, voucher specimens, germplasm, and data. I should have put data on here. Obviously, data is very important to future conservation and provide details for users to be able to select their material. And then lastly, this is my final slide. And the reason for showing you this is not because I'm going to explain to you what it is. It's basically a model we're using in Europe that was developed by the EC-PGR crop-wide relative working group. But it shows that we can do conservation at various geographic levels. So here we're doing conservation at national levels and multiple national levels to develop and interact and be integrated with a European model as well. So we can't ignore the fact that we have all these different layers of geographical layers interacting to do in situ conservation. So final comment, Hawkes in a paper in 1961 said that in situ was in its infancy. It was then. Since 1961, we've done an enormous amount of work to develop the techniques for in situ conservation. And so I think now we are really ready to implement them across the board. And it started. You can see many countries are starting to set up now.
Thank you very much for the very nice talk. I had a question. You mentioned teosinte, which is the wild maize, and there are genetic reserves in Mexico to conserve it. And when you go in Mexico, there are other species or subspecies like Mexicana, parviglumis, which are not yet endangered, but they are becoming rarer. But at the same time, they are considered pests by the farmers because they crossbreed with maize. So they actively destroy it. And as you say, but they like to live in disturbed habitats or in the field margins or edges and so on. How would you do with crop relatives that are considered pests or weeds by the farmers?
That's a very interesting question for me personally. If I can answer from here. Well, let's answer your question first. So if you're dealing with a species that has a broad coverage over a large area, you don't do conservation every single site that has that species. So if you have an area where the locals particularly don't like it, then don't do conservation in that area. Choose somewhere where the locals are supported. The beauty for me of doing in situ conservation and why I find it fascinating is because you're always, every single time, working with local communities. So if you're doing ex situ, really what you're doing is you meet the local community for a short time, you get your sample, and then you move on. With in situ, you maintain the relationship. And for me, that's interesting and exciting. Then why am I particularly interested in what you're doing? Because I was doing a project a couple of years ago in Mexico, and I was told by Canabio that the farmers in Mexico love Chania sinte, and they really encourage it to hybridize. If you're going to get hybrids between Chania sinte and cultivated maize, it strengthens the maize, and that farmers actively plant Chania sinte in the fields. But you're saying that doesn't happen. And let me explain my logic. So I come at this from a scientist, and I looked at this situation. If you have a crop and it's crossed with the wild relative, then what's produced must be less yielding than something that is the original crop. Yes, it seems just but they maintain no. Even in the F1, you get a yield increase. And so I asked for proof of this. I asked them to take me and show me this in the field. In the end, it was never time for us to go to the field to find examples of this. So what you're saying is joy to my ears, because I thought they were wrong. But maybe there are cases where this happens. I can't, I don't have proof that it doesn't. But logically, I don't think it can be the case. I think you're right. I think most farmers don't want this. But then that what they shouldn't then think is that we never want to use crop wild relatives, because obviously if you're doing breeding, you can take the traits out and you're not getting that deleterious. You're only importing a small amount of the genome. Right, Joanna?
Hi, can you hear me? Yes, good. Okay, so now I will, Nigel gave a very good overview about in situ conservation. Now he briefly mentioned about conservation planning, just a few slides, but I will now go into the more detail. So I'll give you a brief introduction. I will talk about what national conservation planning is, how and why we should involve stakeholders in conservation planning tools, and finally linking conservation to utilization. So national, and when I speak about national conservation planning, really can be also at the regional level, can be at any level, but here let's talk about national level. National conservation planning is essential to maintain PGR diversity for the future, because it allows us to identify in situ conservation sites that may form a network to actively conserve priority crop wild relatives or land races or any plant genetic resource, and also to identify sites to collect ex situ sections for long-term conservation and posterior utilization. So in summary, basically we need to identify which diversity of crop wild relatives, land races exist in a particular location or in a particular geographic area, and that relates to the development of preparation of a checklist. Then which diversity is more important to conserve? So then we apply several criteria and methodologies to identify priorities. We can then undertake several type of analysis, diversity analysis, climate change analysis, gap analysis to identify sites for conservation. And then we can obtain these recommendations for ex situ sites and where we should collect for ex situ conservation. So let's go through the first step, the generation of checklists and then prioritization. So basically we need to identify or determine the geographic scope of our work, of our study. Can be a region, which includes several countries, can be a country, can be even a subnational region. The scope of the checklist, whether we want to be comprehensive and develop a checklist of all the resources there, or just the crop-wide relatives, national crop-wide relatives, or even the crop-wide relatives related to a particular crop gene pool. Then we can annotate that checklist with information that will allow us to prioritize that checklist and then make the checklist available to users. And then when defining priorities, Nigel already mentioned some of these criteria. We first find the prioritization criteria, then the methodology, and then we apply both methodology and criteria to our checklists. And we can do it via several means, Access, Excel, etc. This is an example of the work that has been done in the UK. So particularly for developing or preparing a checklist of crop-wide relatives, as you see on the left, as I said before, you determine the geographic scope, so it was the UK. Then whether you want to include in your checklist all the crop-wide relatives of the country or the UK, or just going through a monographic approach, which is selecting particular or priority crop gene pools. Identify a target crop genera, and then we match the crop genera with the flora checklist, and we then obtain a crop-wide relative checklist. Then annotate that checklist for prioritization, and then make available the checklist to users. And in particular for England, Scotland, and Wales, there was this important involvement in stakeholders, and prioritization will also prioritization criteria were identified. So whether the species was native to the region, to England, Scotland, or Wales, the economic value of the crop, the gene pool and taxon group information. So this relates to whether the species is known to be known to be already used in crop gene pool or potential use in crop breeding, whether the species was threatened or not, and then conservation designation. And here in the pictures you can see some of the priority species. And in the scheme in the middle, you have the flow of this process. So the UK flora has almost 5,000 taxa. The checklist of crop-wide relatives then has about 2,100 taxa. And after prioritization, about 223 taxa were identified as priorities. So once you have your checklist, you identify your priorities to conserve, then you start looking at the data, where these species occur, where these populations occur. So you collate that information into a template. You do those several analyses. You can do a hotspot analysis to identify hotspots of these species, or a complementarity analysis, or even looking at, you can even look at the ecogeographic diversity as a proxy of genetic diversity. I will give more details about that afterwards. And then you can also identify the gaps in situ, ex situ conservation, whether the species is actually conserved in situ in protected areas, or if there are any project that is currently managing those populations, and whether those populations are conserved in gene banks or ex situ. And then you can also do, if you want, or ideally, some the analysis of the potential impact of climate change in these populations. And then you obtain your conservation recommendations. So where is priority diversity located? These are some examples I will give you. You can create distribution maps. This is a map of a very small, maybe you can't see, but there are red dots in Portugal from a wild carnation. So a simple distribution map. Besides that, and you should also look at how your data is distributed. So you can see in this is the SADC region, so in Southern Africa. And this map in particular shows the number of herbarium records that were collected in Southern Africa. So the red ones, the red areas, orange areas are the areas that were most sampled. So it gives you an idea of the bias of collecting. Then you can also do species distribution modeling to assess the potential distribution of the species. And here you can see the map for VCA, Ervillia, and Jordan. So the areas in red are those ones where the probability of finding the species is higher. And then you can also combine these species distribution models and of several species. In this case, it was done for the 201 priority crop height relatives in Norway. And you can see that these are the red areas are those with the highest predicted richness of priority crop height relatives. So the other question we want to answer is if there are any gaps in conservation of the priority diversity. So gap analysis is a conservation evaluation technique that identifies gaps in conservation, and it involves the comparison between the range of natural diversity with that diversity that is in situ conserved or ex situ conserved. If you are looking into in situ, then it's called in situ gap analysis. But if you are comparing the national diversity range with ex situ conservation, then you call it ex situ gap analysis. And it can be at any level. It can be at species level, at genetic diversity level, and even at ecogeographic diversity level. So these ecogeographic and genetic diversity is looking into the within diversity, the within species diversity. So it's more detailed and more and provides you with more information. So an example in the SADC region, so in Southern African crop height relatives. So going through that process, this is what we identified. This is the kind of information that you can obtain from the gap analysis. In this particular case, and as we know, I think everywhere in the world, mostly, crop height relatives in Southern Africa poorly conserved, both ex situ and in situ. 50% of the species are not conserved. 50% of the priority species are not conserved ex situ, and those that are conserved ex situ, 40% have less than 5 populations, and 16% have only 1 population in gene banks. And 17% of the species occur outside protected areas exclusively. So current network of protected areas do not conserve 17% of priority crop height relatives of that region. And those that occur in protected areas are not monitored or actively managed. And I think this is a picture not just for the SADC region, but for most of the European countries. So the other thing that you can also do with this type of analysis is to think of, as I said before, is to evaluate the diversity within each species, within each taxon. And you can use, obviously we don't have funds to do genetic diversity analysis for all the species, so we can use ecogeographic diversity as a proxy. Obviously this will work perfectly for some species, and maybe not as perfect as we wanted for other species, but I think it's the best we can do at the moment. So what we can do is to produce ecogeographic land characterization maps, which describe the different environments of the territory or the different potential adaptive scenarios of the species. You identify the variables that are most important that shape the distribution of that particular species. And in this example, you can see a map of an ecogeographic land characterization map for wild apple in Portugal. And this was done in the context of a project where we wanted to target different environments. We wanted to collect from different environments in order to be able to conserve as maximum diversity as possible. This was the aim of this particular approach. Yes. So we used the CapFitogen tools, which is available online freely, and we use these 12 bioclimatic, edaphic, and geophysic variables using this partition clustering around the medoids method. Then, where should we actively conserve priority cropod relatives or priority diversity? You could do a hotspot analysis, identifying where this tax or this diversity is concentrated, but you could also do a complementarity analysis. And this is an iterative process that identifies the minimum number of sites that are needed to conserve all diversity or your target diversity. And it can be based on a grid, or it can be based on a network of protected areas. And also, like the gap analysis can be done at the species level or the taxon level, but also at the within species, within taxon level. And let's, I want to show this example for Zambian cropod relatives. This analysis was done more or less recently. And basically on the left, you see the map of Zambia. And with the different arrows, you can see very, very tiny grids, 13 grids, the 5 by 5 kilometers, complementary grids that will conserve all priority cropod relatives. So for them, they identify 21 that they really wanted to conserve. And these are 13 grids that are needed to conserve these priority cropod relatives. And on the right hand side map, you have the analysis was done looking at the protected areas. So whether the protected areas could actually conserve, the existing protected areas could actually conserve priority cropod relatives. And it was 10 protected areas were identified that would be able to conserve 18 cropod relatives. But three of the priority species occur exclusively outside protected areas. So there's a need to identify 3 additional sites outside protected areas. So how is climate change predicted to affect target diversity? This work was done for Jordan, for Vicia sericocarpa. And on the left, you don't see very well, but you have the potential distribution of this species with current climate, in the middle with 2080 climate, and on the right, which is what I think is more important to us, is the predicted change. So sites or areas in red are those ones that are predicted to disappear completely. Those in blue are those ones that where there is no impact. So the species is predicted to occur there, but there is no impact. Well, predicted to be no impact by climate change. And then you can, I like this slide because it shows you how climate change may affect the distribution of the species. And this is for the SADC priority cropod relatives. You have the winners. So these are the species that actually gain more area than lose. And then you have the losers, which are more than the winners. And the losers are those ones that lose more area than that area that is gained. So this information can also be important when you are planning for conservation. And now, when you put all this together, what we actually want for in situ conservation is to conserve in situ as much diversity as possible, but populations that persist in the future, right? So when we merge all these ecogeographic diversity, climate change analysis, and complementarity analysis, these were the sites that were selected to conserve priority crop out relatives in the SADC region. And if you look at ex situ, the first priority is to conserve that diversity that is not conserved ex situ, and it is likely to disappear with climate change. And second priority is that diversity that is not conserved ex situ, but is regardless of what is impacted by climate change or not. So almost there. It is really important to again engage stakeholders, national stakeholders in planning, and we can do it via periodic meetings, scientific committees, consultations, workshops, because it's really important to raise awareness of the diversity. It's important because we also learn a lot with them, with the stakeholders, and I think very, very extremely important that it facilitates winning over ownership of the work by stakeholders. So it's easier to actually implement conservation actions and identify priorities for conservation, identify the methodology and criteria, first of all. And so we circulated the document before the meeting. So then when we got to, then on that document, we were able to describe the various options. And at the meeting, the methodology and the prioritization criteria were voted. And then here on the left is just to say that national stakeholders in this work in Jordan, and this was to develop a national strategy for socioeconomic plants for Jordan, they were involved at every single step, since the flora checklist, even the red list status, the red list because they were involved in red listing of the species, for prioritization, as I explained in the picture, and then after that, in the preparation of the strategy. This was all circulated and approved afterwards. Conservation planning tools. They are very useful to guide countries for conservation planning, in development of national strategies. They promote standardization of protocols and minimize errors and time costs. And I'm only talking about this one. It's an interactive toolkit for crop out relative conservation planning. It can be used with any plant group, really. And within this one, you can find many other tools. And in this particular toolkit, it includes a good description of each conservation planning, examples, interactive schemes where you can choose whatever suits you better for your national context, and all sorts of references and tools. To finalize, as I showed you this scheme before, and it's just to show again how this continuum between diversity, coming from the diversity available and utilization is important. And among that, between that, you have all these conservation planning tools, conservation planning, the development strategies, and then the active in situ and ex situ conservation and utilization of diversity. And as I said, conservation planning is important to identify sites for in situ and for ex situ conservation. And the national strategies do really help to coordinate this systematic approach of conservation. Oh, I had another one. It's just, I think it's the final one. Let's see. Yeah, sorry. So just to say that indeed we could, using all these tools, we can try to identify where potential traits are found in our accessions, in our populations in situ. We can use this predictive characterization technique, and this was done for the SADC, Wageningen accessions and populations of the SADC region. And the idea was to identify populations that were predicted to be drought and salinity tolerant, and that occur in different ecogeographic environments. This graph shows you gene bank accessions of all Wageningen wild relatives in Southern Africa, herbarium vouchers, and then very small, but in the middle you can see black dots showing the selected samples that are predicted to be drought and salinity tolerant. And what we did was to develop an ELC map, ecogeographic land characterization map, and then select from those previous accessions, that subset, select those that occur in various ecogeographic diversity, various ecogeographic environments. So here you can start linking conservation planning and utilization.
After you establish, and what is the frequency of the observation? Yes. Do you go back to observe? What kinds of frequency? Once a year, or did you go back to? Yes.
I think it depends on the species, right? If it's a tree, it's a long period, but if it is an annual plant, then you have to be more frequent. But I believe, I don't know, I can't remember the every year, probably every 5 years for annual species. It depends even if the species is threatened or not. If it is being threatened, then you have to be more frequent and see what is happening there. If it's completely, if it's free of threats, then you can be a bit more less frequent. It depends on the species.
Any other questions with Joanna? Yes.
Just want to ask, because were these areas kind of protected? Because what I know from Africa, usually those spots are treated by grazing or something like that. No one really, if there is not fence, it's really hard to protect those plants. So if they were protected or not.
Well, in some cases, I actually the top site for Southern Africa, I visited, and it's pristine. It's really, it's a private genetic reserve, and it's pristine. But having said, of course, there was one of the sites was what's the name of that safari place? Kruger, and I don't know how it is, but maybe it's a bit more. There is people coming there, and it's not as pristine as the other one. But I mean, it varies. It varies a lot. There are sites in Angola and Botswana, etc., and it really varies. So I don't know. The only one I know, it's the top site, and it was.
So this is a kind of university to teach a wider audience about how it should be done. Nigel outlined the whole problem of in situ conservation. Joanna subsequently explained how to do it, what all is necessary to need. I decided to take it from the very opposite corner, just to point out problems. Yeah, not describing examples in Africa. I was also engaged in let's say proposing of wild cottons conservation in Africa, which was very interesting. But I will return to the small conditions of our country as a case study and point out problems what we are faced to. So I will my talk will be consisting from two objectives: general problems, and then I will show you cases of different crop groups in Czech Republic, how it is possible to conserve them in situ. We have theoretical optimal case. Crop wild relative conservation is a request from government, or even better from European structures. Conservation is ensured jointly by sectors, agriculture and environmental. Management plan is confirmed by both ministries, national agriculture and environment. all possible stakeholders are nicely collaborating, and the whole system of conservation is fully funded. Maybe the last point is very, very fundamental. But the practice is usually very different. If we look at I put on 4 problems. The first problem is mandate for crop alternative conservation. We have agri sector and envy sector. Crop alternatives are needed for breeding and other uses as a new and very important commodity in agri sector. On the other hand, in envy sector, crop alternatives are not interesting unless they are endangered and protected. However, within these endangered and protected crop species, we have joint interest. Yeah. So envy sector has a very good knowledge of wild plants conservation because they run in situ conservation of protected species. They have available protected areas, usually public lands or government lands. So land ownership is ensured. Funding for land and species conservation is ensured, but definitely not for crop alternatives unless they are endangered or protected. So in agri sector, we have knowledge of conservation, but rather through ex situ, from cultivation. We have some land ownership within farmers and community land or public land. We have also funding for cultivated plants, which cover crop alternatives very, very, very partly. From the talks, you could learn that some countries have environmental and agricultural sector under one ministry, or some countries biodiversity is considered as a whole under envy sector, probably in Netherlands. But most of the European countries have environmental and agronomic sector, agriculture sectors separated with own and very different policies, unfortunately. So again, we need very close collaboration between these sectors. Otherwise, it is difficult to manage conservation. Agriculture sector, I would say, this is my statement. Yeah. So we need to include the term crop relatives into the vocabulary of Ministry of Environment or generally to environmental sector, and we are doing everything for that. Yeah. Unfortunately, we have completely different methodology between these sectors, and we have to agree on some compromise how to manage in situ conservation. So consultation between these sectors is highly needed to find solution for crop alternatives. We are in the Czech Republic. We are in the stage that we have already had several consultations, and still in October we plan to have consultation on the floor of Ministry of Environment. So hopefully it will bring some fruits. So problem 1 is subjects of conservation. They are protected areas, national parks, protected landscape areas, national reserves, and the Prague park rangers cover conservation very nicely, but will not do much for crop alternatives without a special funding from agri sector. But we are happy we have money for national program, and we cannot share this money to nature protection. Farmers and agricultural land keepers can be good custodians of corporate relatives based on mutual agreements. I have many such good experience when I did some monitoring in the country. NGO and other organizations can be also good custodians for corporate relatives, again, based on mutual agreements and available funding. In addition, land under governmental ownership or state ownership, how to say it, it means belts along rivers, roads which are under, let's say, companies caring for roads and trails and so on, and forests. In these cases, there are mostly not responsible people, and custodians of these in situ conservation must be on agriculture sector. Problem 3. Protected areas and environmental policy. Again, I think already mentioned that protected areas can manage conservation properly, but backup is a problem. Backup is a problem because national park usually have so-called no-touch policy, prohibiting any collection for backup. So, for instance, from national park, from first zone, it is impossible to get material for backup to our gene bank. Plants listed in the res lists usually are legally protected and cannot be collected. Another problem. But among those are, of course, desired crop alternatives, because we don't want to take care for common species, and our preference is to take care for species which are crop alternatives which are less distributed or threatened. Environmental sector recently allowed us collecting in buffer zones of national park or protected landscape region, but the required material may not be available there. So maybe I will show you on a practical some example later that it is these cannot cover these species which would be for us the most desirable. Another problem is some crop relatives represent unwanted material for environmental sector. It was already mentioned. There is an urgent need on compromise, and while I was consulting with nature protection, they finally admitted that it is possible to change the Act 114 that is on nature and species conservation. So I was very surprised that this is some kind of breaking of icebergs. Let's see. Problem 4, or longevity of in situ conservation. Longevity can be ensured in protected areas without problems for minimum 10 years or longer. On the other hand, private owners are very enthusiastic in conservation, but signing agreement for a longer period is a problem. Governmental sector can agree, but in case of constructing works, the in situ plots have very low priority, unfortunately. So I will now touch a case study in the Czech Republic, getting out of the problems. As Joanna mentioned, we had this inventory and published strategy for in situ conservation together with Nigel Taylor, who was sent by Nigel to us, and we did the prioritizing for food and feed species. We're using unique, excellent botanical finding database with more than 6 million records of with sites and which are georeferenced. The plotting of relative distribution was done for the Czech Republic. The second prioritizing was for socio-economic importance. So we finally, I jump over all these processes. So finally, we came to 204 species, which are plotted in this map, upper map. You can find the red, the most rich regions, and the final result is the second map, where conservation was planned within current protected areas, plus roughly 8 to 10 additional sites. We also had extensive collecting program since 1993. We collected more than 6,000 crop wild relatives in the Czech Republic, and of course we had this database. It was very useful for this planning. And now I would like to show some practical cases. We have some sites are monitored. Just a few numbers. Diversity of species in Czech Republic. We have 965 genera, 3,713 species and subspecies in the Czech Republic. And in our national program, we keep 1,392 species belonging to 463 genera. So these high numbers are after our collecting activity within Czech Republic. And we keep also rare, protected, and threatened plants, 137 species, and another 182 species we keep as in working collection as service for environmental sector. And now these examples, we selected different crops where we have long-term monitoring. For instance, chive, we have monitoring from 2008. Every year we monitor number of species, and we calculate botanical diversity of the sites, because we have a permanent plot 3 to 3 meters where we make sign when we note phytosociological relevance and calculate Simpson and Shannon indexes, and these are shown in this graph. This is a place, one place of one plot of this chive in Děčín area along Elbe River. Another species is grass, Hierochloa odorata, which is critically endangered. We have 5 sites in along Elbe River on sandy deposits, and this was another candidate for in situ conservation. But unfortunately, succession of vegetation and shading by enlarging trees deteriorate this site, and I'm not sure if it is finally it can be finally accepted. Tritice. This is my subject. Aegilops cylindrica was found in several sites in Czech Republic, and natural distribution ends in southern Slovakia. Unfortunately, Slovakia is not our anymore, so I have to leave it. I have to leave it to my colleague Pavel Hautvogel. But we have Agropyron pectinatum, which is which is critically endangered. There is one site in southern Moravia which we could conserve, but everything depends on negotiation with the agency for nature protection. I had also fantastic site on the bridge in Prague. This is the picture down here. It's a railway bridge where is a population of Aegilops cylindrica for, let's say, 20 years, and I do monitoring. But unfortunately, this spring I went there, and they started to repair the bridge, and they excavated my Aegilops. So I'm very, very upset about it. Wild hops is another important crop because we are a beer country, so it is important. My colleagues from Hop Research Institute, Vladimir Nesvadba, did monitoring of populations in Jeseníky mountains on the Polish border, and he found a lot of possible localities, and it is inside and outside protected landscape region Jeseníky. Their conservation depends on agreement with the agency and with private owners. If it is on private land, these people are eager to conserve it, but they don't want to sign anything. Other candidates, I would like to mention Glycyrrhiza glabra, which was historically planted, and now it is naturalized for maybe 30, 40 years, 50 years in protected landscape region Pozdřany, and it would be an excellent example for conserve in situ. We have also in the same area Cerasus eminens. This is a natural hybrid of sour cherry and Cerasus fruticosa. It is also interesting. We have in vicinity we have flax Linum flavum in Dunajovice Hills, and Nigel sativa is named according to you, which was used in Near East. This is probably introduced in historical times, and it was used for let's say putting on Arabic bread. Okay. Then we have additional in situ candidates in grasses and leguminous plants like Stipa capillata, Festuca gigantea, Poabadensis stipa, other species of stipa, in some in protected areas, some outside. So it is again in work. And concerning very critical endangered species, we have Rubus hamemorus, 20 small population in Krkonoše mountains, in peat box. It is the most southern distribution of this rubus, generally common in Nordic Europe, in Norway. We did monitoring also in Svalbard, but again it will be a problem to collect some samples for backup. And A similar case is Ribes petreum, which is in 3 localities in mountains Krkonoše and Jesenike, and there are only, for instance, 30 bushes in Elbe, Serk, the spring of Elbe River. And again, it depends on a negotiation with the agency. Sorbus sudetica is our endemic species of hybridogenous origin, Sorbus aria and Hamemespilus from postglacial, and it's endemic to Krkonoše mountains. There is, I would say, on-farm conservation of it in Krkonoše Park headquarters, where there are grafted specimens from all available bushes. Again, we don't know how it will be possible to include or not. Okay, and the last, I would like to show our agreement with Agency for Nature Protection. They offered us donor plots for species, for making grass mixtures. So there are quite common grasses and dicots, leguminous species, which we can simply to use for in situ conservation, and we can get several such possibilities for. So this is overview of our problems. Of course, it would be much nicer to talk about Africa and.
Thank you. I'm not yet familiar with the EU laws pertinent to indigenous people's rights, but in many countries are facing this challenge when it comes to carrying out in situ conservation, and that is, how do we reconcile the conflict between prioritizing the hotspots, the genetic reserves, and the rights of the indigenous community, especially when it comes to tenure and land use?
I was talking about our conditions. So I think we don't have problems with indigenous communities in our country. We have basic problems to negotiate the procedure with between sectors and environmental and agriculture. That's all. So with private people, it's possible to negotiate. This is I want to oppose Nigel, because he promotes national reserves, system of national reserves. It would be nice. But now I see that the negotiation with this environmental people would be much more difficult than simply to negotiate it with private owners and for instance along rivers. It's probably very easy to negotiate with these companies.
So the answer to the question is, it's never easy to negotiate. It always takes time, but I think you're right. Generally, apart from the tsunami in northern Norway and Sweden, then maybe there isn't so much of a problem with that. But the area where there is in Costa Rica. There's something called the Guanacaste protected area. If you look it up on the internet, Guanacaste, and it's always cited as an example of what they call biocultural restoration. So it was an area that historically had a lot of biodiversity, and then they started setting up wide-scale meat farms, and then cutting down the vegetation. And then over years, they destroyed the overgrazed, and so they destroyed the environment. And so gradually they started reinvigorating the forest. And they found by reinvigorating the forest, not only did they promote biodiversity conservation, they also promoted the culture of the local people. So the local people welcomed it very much as securing their rights over the land. because it was recognized by the government that they actually own the land and they had no rights over it beforehand. So in that case, it's a very good example of where it works. So if you search for biocultural restoration, Guanacaste Nature Reserve in Costa Rica, then you'll find the story. Thank you. Ready?
Yeah.
Okay. Thank you very much.
Well, thank you, Nigel. I just want to say thank you to Giovanni for the invitation to give this talk. I think it's a good opportunity. And even though we may share different genetic resources as gene banks, I think we share the same ideals and the same issues, same problems. And let me start with a little bit of history of our background. And Arabidopsis is not very spectacular, I have to say that, but it's very useful. It's like a little perfect plant. And as in medicine and in pharmacy, you use mice to experiment everything and try everything. It's the same with Arabidopsis. So basically anybody that is doing basic plant science, crop science, agricultural science will use Arabidopsis at some point. Not only that, it's also being used in ecological studies, evolutionary studies, climate change as monitor, so biofuels, food security. So it's a very useful model in all areas. What makes it very useful is that it's a little perfect plant, so it has everything you need, and it grows very, very fast. So that means that any experiments you do in the lab, you can see the results in just a few weeks. So from seed to adult plant, it will take 6 to 8 weeks. And it's very easy to grow. It grows everywhere. It grows on concrete. All you need is just a drops of water, and it will be fine. So a little bit of history about us. We are located in Nottingham, which is not in London, because I always get that question. And we're basically 2 hours north from London. And we are famous for Robin Hood. But I'm afraid if you go and visit us, that's the only thing we have of Robin Hood, which is a statue this size. And it's more famous now for the other building on the right, which, if you don't know it, that's where the Dark Knight was filmed. So that's Batman's house, Bruce Wayne. So I know it's a lame joke, but it's Batman and Robin. Yeah.
Oh.
I like it. So the stock center started about...
Just 5 minutes on your talk.
No, it's fine, don't worry. 30 plus years ago, when in the 90s it was decided that Arabidopsis should be the model plant. So people online is my joke. So here it goes again. Okay. So we started like 30 years ago with just a few hundred stocks. And then what happens is Arabidopsis is the first plant to have the genome sequence, and it's an explosion of stocks. Everybody starts working on that. So compared to 200 stocks back 30 years ago, and right now it's around 1 million, all of them genetically different. Demand for this resource is so high that one center is not enough. So there was an agreement, and now there's an American center in Ohio State University. The idea is that they will take care of all the American, Canadian, and South American customers. We take care of Europe, and the rest of the world can go to either center. Now, there are 2 centers. They're supposed to have exactly the same stocks, the same collection. And if you are in Europe, it's easier to send the stuff to us first. We'll grow it and send it to the US, and the same with them. So the idea is that there are mirror stock centers. We should have exactly the same. There's usually a delay. And everything we grow and we bulk, we send to the States and vice versa. So in a year, roughly, we will receive 5,000 new stocks from the US, and we will try to send them the same number. So the collection is always expanding. And you can imagine from 200 to 1 million. At the end of the notice, we will receive roughly every year 5,000 new stocks every year. So it's just an explosion. So why do we exist? Well, first of all, because we can keep the stock safe for you. Number 2 is if you're a researcher and you just publish a very important paper and you mention your stocks, instead of people saying, oh, I just read your paper, can you send me these? You can send them to us and we'll distribute on your behalf. And the other way around. If a student or a researcher is looking for a specific stock, instead of going through all the literature or all the search engines, they can come to us, have a look, and if there's anything they want, they just come to us. So it's just to make it easier for everybody. As I said before, 1 million stocks, roughly 95% of them are transgenic, but the rest are natural Arabidopsis stocks that have been collected all over the world, from Cape Verde to America to Central Asia to Africa, obviously in Europe. And now we have started receiving more species. So we have 29 new species, most of them related to Brassica and other model plants. So what's happening is other communities are saying, well, now that you have the infrastructure to do this, will you be okay to receive this collection and then distribute it? And yeah, that's fine, no problem. So there's a mixture of everything. So if you want a stock that has no roots, it's there. That has variegated leaves, is there. That has no flowers, it's probably going to be there. Most of the other mutations are non-visible. So it's changes in the genetic code. So this is just to give you an idea of how many people look at our website in a single day. So it's all over the world. That doesn't mean that they buy anything, but they look for information or for ideas. Yeah. And as I said before, Americans should go to America and the Europe should order from us. So when it comes to the distribution numbers, when I started working there back in 2008, we were sending 50,000 stocks per year. Just before the pandemic hit, we reached 120,000 stocks per year. If I show you the numbers, you will see that Germany used to be #1 every single year. And then around 2010, China came to play. And since then, usually China is #1, Germany is #2. And then you have a mixture of the UK, sometimes it's France, sometimes it's Spain, Switzerland. It usually depends on the stocks. So it could be that one year there were a couple of new donations or new collections that were very popular within the scientific community, and everybody wants to work with that. So for example, at the moment, there's a collection of 1001 genomes, it's called. So it's 1001 different ecotypes that were collected all over the world. And it was curated and it was put together by this very important group, I think, in Sweden, and it's very popular. So we have sent tens of these stocks. And I checked my records, and a Korean guy did order 3,000 stocks for us, and we sent them to them. Yeah. So let's talk a little bit about money. We are funded by the UK government, basically. So at the moment, all the staff that work for us is covered by the grants, which are renewed every 4 or 5 years. That's his job. So everything else, consumables, infrastructure, new equipment. If you think about it, if we're working with thousands of stocks, I need an army of people to be working in the lab or in the glass houses. So everything else is paid by the cost recovery. We charge a small fee. Now, at the beginning, in the 90s, because this was brand new, there were no fees. They were available to anybody. And even today, schools can order stocks from us and they are free. Then at the end of the 90s and the early 21st century, it was a subscription fee was introduced. So you would pay once a year and you could order as many stocks as you wanted. But that was before the big explosion of new stocks. So it became a little bit unsustainable. So we had to introduce a batch fee. I mean, a fee per stock and a batch fee. So that means that the cost of what you're ordering should be extremely low compared to if you wanted to actually create that stock. And that includes the packaging and delivery and all that. Now, before Brexit, only US and Canada needed phytosanitary certification. So we had to introduce a charge for that because, as you probably know, it's an expensive document. After Brexit, we had no choice. I mean, we spent 2 years without charging for phytosanitary certification because we didn't know what the system was going to be. We were not sure what the rules were. But even now, at the moment, we are subsidizing most of that cost because our priority is for you or the customers to receive the seed. And that document, this shouldn't be an obstacle that cannot be overcome. So we charge a nominal fee for the phytosanitary certification, and everybody seems to be happy with that. And because a phytosanitary certification has to be introduced, that means we cannot send stuff by normal mail anymore. It has to be by registered mail. And that usually involves either FedEx or DHL, and we have to charge a minimum fee for that. Every time prices have increased, it's always in consent with our friends in America. So both stock centers have the same prices, obviously. And also we tell the community in advance. So we have 3 main jobs. One of them is everything we receive has to be propagated. So if you're a scientist, a researcher, and you just created a new collection and there's a paper out there, people are going to maybe asking about it and you want it to be ready as soon as possible. So that's the first thing we do. The idea is every new stock that we receive, even if you only have 10 or 20 seeds, we'll try to bulk it and make it available for distribution within a couple of months. Second thing is we need to keep everything safe. And I'll show you a little bit later how we do it. But again, as mentioned by Theo, everything goes to the freezer and to the specialized seed rooms. They are 15 degrees Celsius, 15% humidity. That are medium storage rooms. So when we are processing the orders, that's where we take the seed from. And we always have a backup at -20 in case if anything happens. And our last and most important task is distribution. And that's a challenge at the moment compared to a few years ago. But yeah, we need to make sure that you receive the stocks as soon as possible and as easy as possible. So for that, we have a website, which is very, very simple. And just think about Amazon, but instead of books, is seed stocks. So we have a search page, and you can look for name, for the donor, for a gene code, for any word you can think of, and hopefully you will find it. Yeah? And you will get a list. Now, the information that you see displayed there, we didn't create the stocks. We're just distributing stocks on behalf of the scientific community. So it's the scientists, the researchers are responsible for giving us the right information. Right? So everything you see, every time we add a new stock to the catalog, we always go back to the donor and say, are you happy with this? If anything needs to be changed at any time, just let us know. We'll do it. So you just add your stocks to a shopping basket. You register yourself, you put your delivery address, any special instructions you can put there, and then we'll just send them to you. As a donor, again, we rely on people's willingness to share what they created to us, and then that they wanted to be shared with the community. So we just have a very simple donation form. So if you tell me, Marcos, we have this new collection, I want to give it to you, I will say, just fill this form. It's the basic information that we need to put it on the website, on the database. And then I'll send you an email saying, this is how it looks. Are you happy? It's ready. So it's a very simple process. I think we could not make it any easier for donors, to be honest. So this is just to show a snapshot of the curation model, because if not, my bioinformaticians are going to be cross. So there you go. So. Right. So we have done a few things throughout the last few years to make it easier. So we had an issue with China at the beginning because we were sending the stuff and they would say, no, I didn't receive anything. And in one of the meetings we have, somebody said, the problem is that when you send the package, it's a Chinese postal system that makes the final delivery. And if the address is in English, they're not going to understand. Like that. So we added an option to enter your address in different languages if that would make it easier for you and to make sure that you receive the stocks. Before we had the option of delivery express delivery, because some people were very in a rush to receive the stocks. And we had at different times have different pieces of text translated into different languages. So the idea is that everything you need or you want to make it easy for you, we probably can do it. All you need to do is ask. A couple of things we've done to make the experience more friendly is, as I said before, a significant number of the stocks were collected in different geographical locations. So we did a map, because it could be that you're interested in Arabidopsis plants that are resistant to cold, naturally resistant to cold. So you can look geographically and say, oh, this one in Sweden or this one in Finland. And you can see the exact locations of where they're collected with coordinates and everything. And then we have a social media channel on Twitter where we try to publish interesting things that may be happening around the community. And it seems to be very, very well received. So this is just a snapshot of how we operate. So every week we will print a list of all the orders that we received the previous week. We give them to the staff and we'll go and look in these rooms, like picking up things off the shelf and come back to the lab and then transfer the seeds to small tubes, which are then a triple package to comply with ACGM rules. So you need 3 layers for security reasons. And then they will go to the packing station. And at the moment, it depends where it's going, is what documents you need. But I can explain that later. And this is when we receive a donation. So back in the 2000s, early 2010s, Germany donated, I don't know, probably 200,000 stocks, and they were sending around 2,000 every month, which are those little trays in the first box. So as I said before, we receive them, we enter them into our database. They have a unique number, all of them, a catalog number. They are barcoded, and they're immediately sent to the glasshouse where they are sown. Back then, 6 to 8 weeks later, we harvested the seed, and then the seed, because that's a new seed, that means that that's the seed you're going to use now for distribution, which goes to our rooms. A backup goes to one of the freezers, and the original seed we received from Germany goes to a different freezer. So we always keep the original seed. And because we collected from the glasshouse, a significant part goes to the Americans. So every plant that we harvest in the glasshouse is split into 3 tubes. One for distribution for us, our backup in the freezer, and the American stock. I could spend a day talking about phytosanitary certification. We know all the tricks now, at least from the UK to the rest of the world. It's not easy. It's not nice. It's not fun, but we have to do it. So as I said before, the way the system works is we have to send to the UK lab a small proportion of your order for testing. Once they deem that the sample was clean and it's okay, we are given the permission to print the phytosanitary certificate. The whole process at the moment is taking 2 weeks. When this was introduced, was taking 6 to 8 weeks. So we're in heaven at the moment. Now, for some reason, the UK makes a difference between EU certificates, non-EU certificates. The non-EU certificates, they send them by post because countries like Japan actually want the physical copy. They don't accept electronic copies. So after that, once we have the certificates, we in theory, we could dispatch the orders. But at the moment, every single European country has different restrictions. So it's not only that all of them require phytosanitary certification as per EU law. Sometimes local or national rules are different. So sending stocks to Poland is not the same as it is to France or as it is to Spain. And in most cases, each country will have different delivery options because in France, DHL is not allowed to transport seeds. So we have to use FedEx. And in Switzerland, it has to be the Swiss Post. And Germany has to be DHL. So, yeah. I mean, it's a challenge. It's fun. It's interesting. But the only drawback is that this used to take a week before Brexit. Now it takes a little bit longer because now we have to separate like countries. And you don't want to know about Australia and New Zealand. So leave that. So as I said before, it's one order, one certificate. We cannot use the same certificate for multiple orders. And for some reason, and this is based on EU law, they only last 14 days. So we have a very small window from when the order is complete and when we receive the certificate to actually dispatch the order. The stocks have to be in the border by the time the 14 days are complete. Otherwise, they will return it. I mean, I don't know the difference between the 14 and the 15 days, but those are the rules. We have the target of processing everything and dispatching in 3 weeks. As I said before, we are charging a nominal fee for the certificate at the moment, and we have different options for delivery. So I think now, compared to 2 years ago, we cracked down the system. It's complicated, but if you tell me a country, I'll tell you how to send it. That I can do. So just a quick look at our facilities. We have a main lab, a distribution lab, with wonderful, beautiful, bright orange lab coats. So Guantanamo, exactly. So we have 3 glass houses. We have a massive one, medium one, a small one. The whole capacity for them in total is 18,000 plants, in all of them full. And we have the goal to fill them 3 times a year. So that will give us around 25,000 plants a year. Now I need to tell you about this. So at the end of 2021, a postdoc student got in touch with us from the university and said, I'm testing these new speed breeding lights for my project. And all the plants that I've grown under these, including different crops, grow really, really fast. I have reduced the life cycle by a third at least. Would you like to give it a try? So, okay, we'll do that. So we did a trial for all the end of 2021 and the first few months of 2022, and the results were amazing. So if I told you that in the first slide, Arabidopsis under natural conditions grows to full development in 6 to 8 weeks, with these slides, it only takes 3 to 4 weeks. So normal conditions, the seed will take 6 to 7 days to germinate. Under these slides, 3 days. So in 4 weeks, we can have a full, complete plant ready to harvest, which is amazing. So that has helped us a lot because we can increase the number of plants we are doing. And if there's an urgent request, hopefully in just a month and a half, it will be ready compared to 2-3 months before that. Now, they're very expensive. So only the little glass house is fitted with that. We try to do it in the other glass houses. Well, the small glass house and our growth room. So anything urgent, anything special, we can grow it quickly and it will be ready in just a few weeks. And they've been very, very popular. So we had people come to see us to see, I hear about your lights. Can I have a look at them? Can you tell me? We did that in the international meeting in Japan, and they were very successful, even in the UK. So we told our American friends. So that's the glass house capacity. For storage, we have 6, 15 degrees, 15% humidity rooms. Most of the stocks are organized in trays, numerical order, barcoded, in carts. Some of them are in tubes now because of lack of space. So all of those are distribution stocks. At the same time, we have 3 walk-in -20 freezers. And the idea that everything you see there should have a frozen stock. That's the plan.
3 minutes.
Perfect. So almost finished now. The 3 things that I'm very proud of is, number 1, we always involve the community in everything we do. They know what we do, and everything we do is to make their life easier. We are non-for-profit, so we don't earn anything. So we just, again, my main priority is for you to get the stocks because your research depends on it, your paper, anything like that. Introducing the fee model has been very, very successful because when it comes to asking the government for money, we only ask for the basic. Everything else we can cover ourselves, including outreach, consumables, temporary staff, speed breeding lights, anything like that. And again, we try to do everything we can to make it easier for the customers. And the last slide is to show you that Arabidopsis is not that boring because we had requests to use Arabidopsis in art exhibitions in Italy, in Scotland. When the Jubilee for the Queen happened, they asked us for seats for the gardens. We couldn't go because there were no tickets, but they were used in the Royal Gardens. And also it's been to space. They asked us for seats to send to the International Space Station and for space shows, Royal Chelsea Flower Show and others across Europe. Our funders, our staff, past and present, our friends in America, because we need to be together, and to the whole plant science community. Thank you.
Perhaps we've got time for a few questions, so I'd like to start off with 2. So how many staff do you have? It sounds like you need 1 million. And then, is it confidential, or can you tell us how much it costs? If you're paying per accession, how much it costs to send that accession?
So when it comes to staff, we have 6 people permanent staff. And then depending on the time, the bulking of the harvesting cycle, we could have another 5, 6, or 7. If we have to fill the glasshouses in one go, we'll get 5 or 6 or 7. If we have to harvest the glasshouses, 5, 6, or 7.
And they're casuals.
Yeah. So average, I would say 10 people at any time.
Cost?
The cost at the moment, each stock is £10. Actually, it was raised to 11. The ADRC decided to change it. Yeah. Okay. So it's £11 plus the certificate, which we're charging 20. It costs over 100, so we're helping with that. And delivery, the university has deals with all these delivery companies, so it's another 10 to 20 pounds. Yeah.
Thank you. Yes.
Thank you. Interesting. I've worked with Arabidopsis before, and I don't remember where we got our stocks from, to be honest. But I had a question about this phytosanitary certificate. So what are the diseases that they're actually screening for? And do I understand correctly that whenever I would order a stock, you have to send samples for screening?
Yeah.
So you cannot do this kind of on the bulk?
No. So the way the system works in the UK, they have different kinds of certificate depending on the kind of testing they have to do. So for Arabidopsis, because this has no commercial value, is the most simple test. So they only have look for basically bugs or any contamination. They don't look for any specific diseases. So just physical check to make sure that there are no fungi or bugs or anything like that. And yes, the rule is we have to send 5%, random 5% of your order to the lab. So for every 20 stocks you order, we have to send one random one. Less than that is one, more than that, I don't know, whatever the number is. Yeah, that's the rule. And yes, you cannot do it by bulk. It's one order, one certificate. There's the idea in the future that hopefully we can be audited and we are given some kind of approved plant exporter or something like that, which means that we can imprint our own certificates after being approved by DEFRA or the UK Plant Health Lab. But if I may say this, they didn't know how messy it was going to be or didn't know what to expect after Brexit. So
the species do you carry and what were the difficulties you encountered in reproducing and in multiplying the seed?
So I think most of the collection is Arabidopsis thaliana, and we have probably another 10 or 20 Arabidopsis and other brassica species like Thelaspi. Now, you're right, some of them are different ball games. So, for example, the ones that are brassicas, Arabidopsis is this size, these ones are this. So it requires a different strategy when it comes to bulking. And some of the Arabidopsis, for example, need to be crossed or are sterile. And not only that, some of the stocks might require different additions to the growing medium. So for that, we have a small grower, and anything that is special like that will be there because it requires special attention. Everything else grows fine. But those that need some kind of manual activity or addition or keeping an eye or high humidity or adding heavy metals to the soil, which is another species, we put them apart.
Okay. And does the fee that you charge cover just which part of your cost does it cover? Does it cover, I don't know, permanent staff salaries? Does it cover equipment and building amortization? Does it cover everything or just part of it and the rest is subsidized by the government?
In general, only the salaries of the permanent staff, 5-6 people, are covered by the government.
Okay. And
everything else,
the building and amortization.
Everything else, consumables, you know, the millions of pots, millions of tubes, the army of people that work in the glass house, traveling, outreach, new speed breathing lights, new compartments, improvement of the seed rooms, everything is covered by the fee, basically.
So you're very cost efficient, I would say.
Yeah.
Okay. Very.
Thank you for the presentation. Very, very interesting indeed. You didn't mention anything about legal stuff. Are you aware of the Nagoya Protocol?
I'm going to leave it to the expert in Nagoya Protocol.
Yeah.
So you, oh yeah, okay.
Yeah, so obviously you became aware of Nagoya Protocol through UK PGRG, several members here, and we have, it's kind of an interesting conversation in terms of the way that Arabidopsis will fit into the Nagoya Protocol. For starters, for example, most of the stocks are generated in countries that do not declare sovereignty over Arabidopsis, so from the UK, from Germany, from America. The only ones that could potentially be covered in strict terms from Nagoya would be the ecotypes have been collected, and nearly all of those have been collected before the cut-off date for Nagoya. So those are kind of things we've taken into consideration. There's also the question of use. So as defined under Nagoya, the kinds of use that these are being put to are not direct use in terms of commercial application. They are academic use, they are publication use, which I know does have it does apply, but in terms of, sorry, did you want?
Scientific use is also.
Yes, but it's not use that generates any income. Right, but the benefit in kind from Arabidopsis is generation of publication, not generation of end product. So I was going to go through the various things. So what we've done is we've gone through and done an assessment of all the different priorities within Nagoya and responsibilities from Nagoya, and tested them against Arabidopsis. And so we give people that document, which you can pass on to if you like, so you can see and judge it, and maybe other people that are familiar with Nagoya can look on it. And we've put that through and brought it up with people at DEFRA, in terms of whether they think that it complies, in terms of access benefit sharing. So does that cover what you want to know, or is there more? Yeah, so the stuff that we have is any legal requirements are covered by, for example, patents that may apply to genes that are used within them. So the biggest issues we've had are in terms of the transgenics. So some of the transgenics, for example, have cauliflower mosaic 35S promoter, which has patents associated with it. So people our users are aware, because nearly everything is transgenic, that the use of them, we don't give them without, they have to take into consideration existing patents on genes that may have been put into these as transgenics. So we had that in terms of a number of different constructed lines, so enhancer traps, promoter traps, activation tags, and various other tools. There are genetic constructs in there, and some of the companies that have been involved in the generation of those materials have passed through exemption for ABRC, for example, and us in terms of passing them on, because the recovering of them from the plant is going to be more difficult for the user than actually using them and buying them from the original company. So it's not worth their effort to track that down. Yes. So the question was, do we use an MTA? So we used to use MTAs, and the problem we had with them is that a lot of the MTAs were associated with particular states. So, for example, the original SALT lines had under the law of California, and people in New York wouldn't sign that. Certainly not some people in other countries. And we had a very large population from Germany, very large population from Singapore, and those had issues with the MTAs being associated with the countries of their origin. And over time, what we've had is we have essentially we've gone for an academic disclaimer rather than an MTA, because there are no rights passed through to us. The donors provide the materials. We are a pass-through centre. They receive the materials, and any legal, as I say, patents associated with those stocks are associated with the materials that have been put into those plants by the originators. So we don't have any MTA in terms of that people have a responsibility to tell us what they've done with the plants. And because it's Arabidopsis, and because the purposes of it are different to, for example, if you're using tomatoes or some other commercial, or for example, something that applies to Nagoya, something taken a new accession from the wild, it has a slightly different approach to it in terms of the research applications. Yeah. Sorry. TMI, right?
It's 6:15, so if you don't mind, I think we'll stop there. But personally, I really enjoyed this afternoon's session. I think it's the core, certainly Theo and I would agree, it's the core of the new RI in going forward. So I hope you've all enjoyed it, and you'll see the presentations, as I said, I hope will all be made available, and you'll be able to catch things. And unfortunately, the screen here, I don't know why, it seems to be much smaller, because I checked all my slides, for instance, beforehand to see if they're readable, and they were, and here they're not. So